Siblings born to the same set of parents can look so different from each other. A mother having A blood group and father having B blood group sometimes give rise to children that have O blood group. How does that even happen? Parents that have a darker complexion sometimes have children that are lighter complexioned and vice versa. How do these characters really get passed on from one generation to another? Well, you will uncover answers to all of these questions and more in this session. Hello and welcome to PW NEET English YouTube channel. I'm Shalini Ma'am, your botany teacher, and in this lecture, I'm going to take you through the chapter Principles of Inheritance and Variation in one shot. As you might already be aware, this chapter carries high weightage in your NEET exams, and in order to help you in your preparation, I'm not only going to share class notes but also certain DPP questions for you to practice. You can access both of these on the PW app if you can join the free batch called Complete Botany by Shalini Ma'am. The link to the batch is attached in the description below. Please go ahead and explore the PW app because it is a one-stop solution for all the resources that you will need in your NEET preparation journey. Now, without any further delay, let's get started. See, when you look around and observe nature, you will notice how every time an organism is reproducing, it will give rise to offspring or next generation, which looks very similar to the parents, right? For example, if you consider animals, when a koala bear reproduces, it's going to produce another baby koala bear and not a baby elephant. How is it that the characteristics of the koala bear, the kind of ear that it has, the kind of snout that it has, how do those characters really get passed on to the next generation? Likewise, when you consider plants, when you sow a mango seed in the soil, you end up getting a mango sapling and a mango tree, which is very similar to the parent plant with respect to its kind of root system, the kind of leaves it produces, the arrangement of leaves, and all of that is going to be very, very similar. It's going to be identical in characters, right? So, how is it that these characters are getting passed on from one generation to another? That's exactly what our focus is going to be in this chapter. We will explore how exactly characters get passed on from one generation to the next. Now, this is the first chapter in the unit called Genetics and Evolution. So let's begin our discussion today by trying to understand what exactly the term genetics refers to. Genetics is a branch of biology similar to how you have cytology, anatomy, morphology, physiology, and all of these other branches of biology. Genetics is also a branch. Like morphology deals with external features, anatomy deals with the internal structure of living organisms, genetics deals with two basic processes. One is inheritance and the other is variation. So, genetics is that branch of biology that deals with inheritance as well as variation of characters from parents to offspring. Okay. Now, we need to understand what inheritance means and what variation means. Inheritance, you may have heard people talk about inheritance generally like that in general English, that this neighbor of mine, she inherited this much money from her ancestors, and this person that I know, they inherited this piece of land from his father, and this other person that I know, they inherited X amount of gold jewelry from their grandmother. So when we generally talk about inheritance and use it in normal daily life, we essentially refer to inheritance as a process by which something is getting passed on from one generation to another. It could be land, it could be money, it could be gold jewelry, whatever. Okay, so that is how we generally associate the word inheritance with. But in the context of genetics, we're talking about inheritance not of material stuff, but of characters. What is that process by which my characters are going to get passed on to my next generation? How is my color of hair getting passed on to my son? How is the color of skin getting passed on to my son? These are my characters, right? The color of our eyes, the texture of our hair, all of these are our characters. We have many such characters. Different organisms have their own characteristic features, right? So how are these characters from one generation getting passed on to the other generation? The study of that process is what we refer to inheritance. So genetics basically deals with studying about inheritance, and at the basic cellular level, at the molecular level, what exactly is happening, how are these characters getting passed on, that's what we're going to study about. Like for example, you can see in this image, there is the dad wearing a horizontal striped shirt and the mum is wearing like a vertically striped shirt. But if you look at the baby has a mixture of both the characters, right? It's not entirely like dad, or it's not entirely like the mum. So children will get characters of both the parents, but they're not entirely resembling either of them. So that is what inheritance is all about. And in fact, inheritance is the basis of heredity. When you see people in laymen talking about why somebody has curly hair, you might have heard them say, oh, that runs in her family, that's heredity. Oh, why is this person so tall? Oh, that's heredity. That's hereditary because everybody in their family is super tall. So you may have heard that. In this chapter, you're going to explore in detail how exactly that gets passed on from generation to generation. Okay, so that's about inheritance. I mentioned how genetics is about inheritance and variation. We now know what inheritance is. Let's try and understand what variation is. Variation is also a very general English term that everybody is aware of. Variation basically refers to variety or difference, right? Variation means difference. So, when you look at families, even when you look at siblings that are born to the same set of parents, you will notice how they can be very, very different from each other. For example, my brother and I, we're born to the same set of parents. My brother is super tall. I'm not like so tall. I'm like average height. And then I'm kind of lighter complexion. My brother is darker in complexion. So even though we're born to the same parents, there is difference between siblings. And there is also difference between your generation and your previous generation. We don't look identical to our parents, right? We do have some shared features. In some ways, you might look like your mom. In some features, you might look like your dad, but you're not entirely like mother or like father, because during reproduction, variation is brought about. The difference that you can see between one generation to the next is what we refer to as variation. Okay, so variation is the degree by which progeny differ from their parents. Progeny means what? The next generation is the children or the offspring. This is not true only for humans. This is true for all living organisms that are sexually reproducing. We're not going to be identical to our parents. There will be some difference at least. Okay. So, here is a beautiful family. There are two parents and there are three children. You can see how the children don't look alike with respect to their features, and none of the children look identical to the parents either. So, that is what variation is about. So, if you look at the name of the chapter, it is called Principles of Inheritance and Variation. We now know what inheritance is. It is the process by which characters from one generation is going to get passed on to the next generation. We know what variation is. Variation is the degree to which we differ from our parents, or one generation differs from the next generation. That is what variation is. So here we are going to explore what are the rules that govern inheritance. Are there any rules, any principles that govern how exactly characters are getting passed on? How exactly is variation arising? So we will learn about all of those principles in this chapter. Okay, so let's get started with how humans got intrigued about genetics and all of that. Of course, when we talk about genetics, we initially start by talking about Mendel's experiments and all of that, but Mendel was not the first person to kind of be, intrigued by genetics and study it. Long, long before humans being the inquisitive organisms we are, they were curious always about nature, and being the smart people that we are, we always try to make our lives easier and use nature for our benefit. So back in the day, like several thousand years ago, according to NCRT, 8,000 to 1000 BC, that is around 10,000 years from now. Okay, so 10,000 years ago, think about that time. Okay, so back then humans were just shifting from being hunting gathering people, hunters gatherers, to settling down. Okay, so when they started to settle down, they were starting to rear animals for food, for milk, and also start agriculture. Back then, even 10,000 years ago, humans were smart enough to know that in a population, variation exists. Okay, like I mentioned, variation is not restricted to just us. You look at plant population, you look at any animal population, within a species, variation does exist. If you take a group of plants, rice plants, let's say, they're trying to grow rice plants, they're doing agriculture, they're growing rice. Some rice plants give better yield than the others, so there's variation there with respect to how much yield is given. And some plants probably are more resistant to microbial infections than the others. Some plants are more pest resistant. So within a population, you can find variation, just like how in human population there is variation with respect to our height, our skin color, hair color, and all of that. Even within plants, if you may not be directly able to make it out, but with their characters and how much of nutritive rice grains it can produce and all of that, there will be variation in a plant population as well. Likewise, if you consider the livestock that they're rearing, they could be rearing cattle like cows and buffaloes and all of that. So even within that population, they knew there is variation. Some cows give higher milk yield compared to the others. Some bulls, they work better and harder in the crop field than the others. They are tougher. Okay, so they knew there is variation existing, and they also knew that this variation was because of sexual reproduction. So, being the smart people they were, they utilized it in their favor. What they did was they allowed selective breeding. I mean, they made sure that they wanted their combination of characters, desired combination of characters in the offspring. So they performed selective breeding. So if there's 5 cows, of which one cow yields like very high quality, high quantity of milk, they specifically selected that, and they selected a bull that would work really hard in the field. It was tough and hardy. They selected that. Both of these characters the humans desired, and they artificially allowed them to mate. Like, not artificially allowed them to mate, they selected these two and allowed them to mate. So in the offspring, again, there would be variation. So they would select whichever one had their desired combination of characters, and they did this for several generations. They did the same with plants as well. So from then, we have like basic idea that variation arises because of sexual reproduction, and humans have tried to use that to their benefit. And that is how we've received. We've gotten many breeds of cows that are high yielding varieties. Like in Punjab, we have a cattle breed called Sahiwal. So these cows are high milk yielding breeds of cows, and they obtained this because of selective breeding itself. Okay, so let me repeat. Humans knew from 8000 to 1000 BC. BC means starting from 1, 2, 3, 4, 5 AD. Like right now, we are in 2026 AD. So 1000 to 8000 years before this, before the starting point 1 to 3 is 10,000 years, right? So back then, 10,000 years from now, roughly, around that time, people knew that one of the causes for variation was hidden in sexual reproduction. So they kind of tried to use it by selectively breeding animals and select among the offspring for those organisms that possessed desirable characters. That is how, because of artificial selection, Sahiwal cows in Punjab have been obtained. Okay, so this is like the early history. But when we talk about genetics, the foundation of genetics was laid by this person called Gregor Johann Mendel. He is one of the greatest biologists. And in fact, he didn't really study biology that much. He had studied mathematics. He was good in physics. And eventually his genetics drew his interest, and then he started working in the field of genetics. So basically, this person was a priest, and he lived in a monastery, and right next to his monastery there was this garden, and that's where he performed all of his experiments. Okay, so he worked for 7 long years between 1856 to 1863, that is 7 years in total. And throughout the course of these 7 years, he continuously worked with pea plants. Okay, garden pea, the scientific name of that is Pisum sativum. And based on his observations from the experiments that he performed for 7 long years, he proposed certain rules or principles which we today call the laws of genetics, or we call it the laws of inheritance. Because of his contribution in the field of genetics, today he is regarded as the father of genetics. What did he do? Basically, at a time when people used to study biology, mostly morphological features and anatomical features, he kind of tried to incorporate mathematics into the field of biology because he was good at math as well. When he performed his experiments, you know, he had a very large sampling size of plants. What do I mean by sampling size? We know he worked with pea plants. It's not like he performed experiments with 2 pea plants and based on his observations, he he went ahead and published his research paper and said, okay, see, this worked for these two pea plants. This is a rule. This will work for every single organism. That's not what he did. He worked simultaneously with hundreds to thousands of plants. The number of plants or the number of organisms you're working with is called the sampling size in an experiment. So his sampling size was so huge, like he had kept working with hundreds to thousands of plants at a time. So because of the large sampling size, whatever observations he got, whatever data he, presented, that had a very great credibility. What does credibility mean? Credibility is like being more believable because it's consistent. Okay, like for example, you all have been studying since the last several years. Let's say you just you were a topper in second grade. You got like full marks in all of the subjects in second grade, and after that you've been average, and sometimes you've failed. And now you can't call yourself a topper because consistently you've not shown that track record, right? Sometimes you failed, sometimes you've done just average, sometimes you're below average, sometimes maybe you're above average. If you call yourself a topper, then it's not really valid because it's not throughout. You're a topper for second grade in your second grade, but not anymore, right? So it's not believable, it's not credible. What happened with Mendel was he had a very large sampling size. The reason is because he knew statistics very well. He applied statistical analysis as well as mathematical logic to understand how exactly inheritance happens. In statistics, there's this one law called the law of large numbers. Okay, I'll give you a very simple example. See, when pollination happens, when pollen grains go and drop on the stigma, land on the stigma, there are mighty chances that the pollen grain carrying the tall allele or the dwarf allele can go and fall on the stigma, or the pollen grain that is carrying the information for round seed shape or wrinkled seed shape can go and land. It's a random event, and nobody can really control which one went and sat there, got deposited on the stigma. So it's entirely random. But still, we see how consistently Mendel gets the ratio of 3:1 or 1:2:1 in the monohybrid cross, and 9:3:3:1 in the dihybrid cross. How is that? How is that uniform? How is that maintained every time we perform that experiment? See, we are going to believe these results because it's believable, it's credible because of his large sampling size. Whenever there is any process, any phenomenon where there are two probabilities, okay, for example, like flipping a coin. You flip a coin, there's 50% chance that you will get head or you will get a tail. So if I do it 2 times, both the times I might get head. If I do it 10 times, I might get 4 times head and 6 times tail. If I continuously do it for a number of times, the more number of times I do, the closer I'm going to get to 50% head, 50% tail. Okay, so whatever is the actual probability, 50% head, 50% tail, that I'm going to almost achieve if I keep repeating it for a number of times. By the time I do it 100 times, I may have got 30% head and 70% tail. If I do it for 1,000 times, I might get 40% head and 60% tail. The more number of times I do it, I repeat the same thing. Eventually, with the number of trials, the probability of it becoming 50% is going to increase, right? Same thing happened with Mendel's experiments as well. Since he had such a large sampling size, his results had greater credibility. It's not like one time you get one ratio and another time you get another ratio. Of course, he got differences, mild differences. But the more number of times you perform the same experiments, those differences kind of cancel out and you come to like one common ground. Okay. So that makes it more credible. Now, he not only tested these results on one generation and said, okay, come on, this is what I got. And everybody should believe that this is what is going to happen. This is how the results are going to be for all of the plants, for all of the organisms, for every character. That's not what he did. Whatever results he got, he wanted to check if what he's thinking about those results, what he's inferring from those results are true or not. So he kind of repeated the same experiments with successive generations. So when he did that, he kind of got similar results, and that gave him more, proof that his ideas about inheritance are really true. And only after that he published his articles. Okay, so his data was very much credible because of the large sampling size and also because he repeated them in successive generations, and he got similar results. Okay, So that's the reason this guy is called the father of genetics. Now, let's try and understand why Mendel specifically chose the pea plant for his experiment. There are so many options. It could have been a mango tree, a banyan tree, or any other animal, but he specifically chose this because it has certain advantages. What is Mendel trying to do here? He's trying to learn about how characters get passed on from one generation to another. It's important that the organism we're using for the experiment has a short lifespan. If it has a short lifespan, a short life cycle, then very soon it's going to give rise to the next generation. It's going to reproduce quicker. If you take a mango tree, for the mango seed to grow, form a sapling and become a tree, produce flowers and give rise to the next generation, is going to take like a really long time. So it's going to be too time consuming to study two generations. But in pea, that is not the case. It has a very short lifespan. It is an annual plant. Within one season itself, it will produce flowers, fruits, seeds, everything. So we're going to get the next generation very quickly, which is exactly what we want, right? So because of its short lifespan, is advantageous. That is one of the reasons why he chose the pea plant for his experiment. Secondly, now the pea plants do not require too much of maintenance or labor. They are small climber sort of plants with weak stems, and in a very small space, it's possible for us to grow many pea plants. Okay, so that is the second point. Thirdly, within a pea plant, you can see a variety of visible characters with contrasting traits. Now, what do we mean by visible characters and traits? We'll understand that. For example, in us, the color of our eyes, the texture of our hair, how tall we grow, the color of our skin, all of these are characters. And all of these characters vary from one person to another, right? The color of the hair could be black, it could be brown, it could be blonde, it could be red. The color of our eyes could be black, could be brown, could be, blue, green, hazel, whatever. So each character has different variants and those are called traits. Like that, even in pea plant, there are many characters which have alternating forms that we call traits. Like skin color is a character. Dark brown, light brown, fair, dark, that is traits. Hair color is a character. Whether it is black, brown, red, blonde, that is trait. Like that, even in pea plant, there are many characters that have different traits, and these traits are contrasting. Contrasting means it's not like dark red and light red. They are also traits, but there's not too much of a stark contrast. But in pea plants, there are many characters that had contrasting traits, which means they are very evidently, the difference is evident with your naked eye itself. Either it is like growing super tall, 6 to 7 feet in height, or it's just going to be 1 to 2 feet in height, or it's going to produce violet flowers or produce white flowers. To distinguish between violet and white, you do not need complicated instruments, right? You look at it and you will know. To check whether it is tall or dwarf, you do not need any complicated equipment. You just look at it and you know. Such visibly contrasting traits, many were present in the pea plant. We are trying to understand how characters are getting passed on. Many such characters were there in the pea plant, which could be easily studied without any, sophisticated equipment. So that is also one of the reasons why he chose this plant. Next, the pea plant, once it reproduces. How do pea plants reproduce? By producing flowers, because they are angiosperms, right? So once flowers are produced, eventually the fruits will form and the seeds will form. From one plant itself, you're going to get many seeds. So these seeds are like the babies for the pea plant. So from one parent, going to get many offspring. I've already mentioned how the more number of offspring that are there, the more is the sampler size, greater is the credibility of the data that we get. So from one round of reproduction itself, you're going to get many progeny plants. So it's easier to study how characters are getting passed on. And pea flowers, these are bisexual. They have both androecium and gynoecium. If you allow it undisturbed, it's going to undergo self-pollination, but cross-pollinating them is also easy. Okay, so because of that, he chose these pea plants as his experimental organism. Now, what exactly did Mendel do? There are many characters in a pea plant, just like there are many characters in us, even in pea plant. The shape of the leaf, how wide the leaf will grow, how is the leaf attached, what is the position of the leaf, like what is the phyllotaxy, and what kind of root system. All of these are characters of plants. So all of these are characters of pea plant also. In pea plant also you have all of these characters, right? But there are some characters that show contrasting traits. In all pea plants, the root system is going to be taproot system because it is a dicot plant. In all of them, you're going to find reticulate venation because they are dicot plants. These are characters of the plant, but there is no variation in that. They are fixed characters. There's only one type of character you can find in these characters, right? One variant in these characters. But there are some characters which has two variants, okay? For example, how tall the plant can grow. Either it can grow very tall, 6 or over 6 feet in height, or it can be under 2 feet in height. This is a contrast. The character is one character, which is the height of the plant, the height of the stem. It can grow very tall or it can be dwarf. So there's a contrast here. It's not like all pea plants are tall or pea plants are short. So within the pea plant population, this character is showing variation. So the two variants of that character are called traits. There could be characters which have uh, which is similar in all pea plants, like the ones that I mentioned, the taproot system and reticulate venation. However, there are some other characters which shows two distinct variants that are easily observable. So he first observed and made a list of all of the characters in a pea plant that showed some amount of variation that he could use for his experiment. In some of those characters, initially he selected a lot of characters. In some of those characters, he saw that some characters were influenced by the environment. How wide a leaf can grow can be influenced by the environment. And many other characters had more than one alternating form. So he just did not continue with those characters. Ultimately, he finalized 7 characters. In all of these 7 characters, there were 2 contrasting traits. So he totally chose 7 characters and 14 contrasting traits. So he investigated characters that were manifested as opposing traits. Okay, each character should have 2 possibilities and there should be a stark contrast between them. Such characters he chose. Now, what is a character? What is a trait? These are terms that confuse a lot of students. Let's try and understand what a character means. Like I mentioned, a character is the color of the seed. The color of the seed could either be green or it could be yellow. These are the traits. Okay. So we call these the contrasting traits because it's not too difficult to distinguish between what is green and what is yellow. If this was light green and dark green, it is a trait. There are 2 variants, but it's not too, there's no stark contrast between them. It's unless you compare them, you can't say which is light green, which is dark green because the difference is very minimal, right? But here in the characters that he selected, the traits were visibly contrasting. Okay, so that's one thing. That is one example. Another example that you could take is the color of the flower. The color of the flower could be violet or it could be white. Again, it's easy to distinguish. There are only 2 alternatives and the distinction between 2 of them is wild, right? So very easily you can observe it. So these are traits. This is the character. These are the traits. In all of the characters that he chose, he made sure that every character had only 2 contrasting traits and he chose 7 of those characters. Okay. So which are the 7 characters that he chose? Let's take a look. One character is about the stem. 2 characters are about flower. 2 characters about fruit. 2 characters about seed. 1 stem, 2 flower, 2 fruit, 2 seed. Okay. Let's talk about the stem character. It is the height of the stem. How tall the stem can grow. If it can grow to a minimum of 6 feet, then it is considered to be a tall pea plant. If it is shorter than 2 feet, then you call it the dwarf plant. Pea plant exists only as dwarf, 2 feet under or 6 feet over 6 feet. You don't find pea plants that are 3 feet, 5 feet, 4 feet. There are no in between here. There are no intermediates here. Either it is this short or it is this tall. So this is the stark contrast that I was talking about. Okay, there are only 2 variants and it's very easily visible to your naked eye. That is about the height of the stem. Speaking of the characters related to flower, one character is the color of the flower, the other character is the position at which the flower forms. The color of the flower can either be violet or it can be white, which is obviously a stark contrast. And the position of the flower, whether it is getting produced at the terminal tip end apex of the plant, or it is getting produced at the axil of the leaves. So these are the two variants. If it's getting produced at the axil, you call it axial flower. It's getting produced at the tip of the plant at the shoot apical meristem, then it's called the terminal flower. Okay, terminal position. Speaking of the two characters related to the fruit. The fruit in a pea plant is called the pod. Okay, so the pea pod's color, the pea pod's shape, these are the two characters. The pea pod can either exist in green color or in yellow color. The shape of the pea pod can be inflated like a balloon into which you've blown air, or it can show certain constrictions. So it can either be inflated where it is fuller, or it can show constrictions like this, in which case it is called constricted. Now speaking of the seed characters, the seed's color and the seed's shape, both of these are characters that he considered. The color of the seed could either be yellow or it could be green, and the shape of the seed could be round or it could be wrinkled. So totally there are 7 characters. Of the 7 characters, one is of the stem, 2 are related to the flower, 2 are related to the fruit, and 2 are related to the seed. It's important for you to remember what are the characters, what are the contrasting traits. Okay. Now, what exactly did he do in his experiments? And all of the experiments that he performed, he followed 3 simple steps. Okay. The first step is the selection of parents. When he selected parents, he made sure that the parents were showing contrasting traits. Okay. So in the first generation, in the parental generation, he made sure that one parent was tall, the other parent was dwarf. If one parent had violet flowers, the other parent should have white flowers. If the one parent produces seeds that are yellow in color, the other parent should produce seeds that are green in color. So whichever character he chooses, he made sure that in the first parental generation, they are displaying contrasting traits. Not just that, he wanted pure lines or true breeding types in the parental generation. Okay. So, what are these true breeding lines? I'll tell you. True to its type, we say. Like, for example, you take a tall pea plant. You should take a pea plant, tall pea plant, in which you're sure that all of its offsprings will be tall. It'll be true to that character. That's what it means. So a tall pea plant, if it ends up having 1 or 2 dwarf offspring, then that's not true breeding. If it's true breeding, then essentially every offspring that it produces should be of the same type as the parent. If you have a dwarf plant, then all of the offspring that the dwarf plant will produce, if it gets self-pollinated, all of the offspring that it produces, it should be dwarf only. It should follow the same trait. It should display the same trait. It should not display the tall trait. Okay, so that is called true to its type. If a plant is producing violet flowers, if you allow it to undergo self-pollination, then it should produce offspring which will all produce violet flowers. Such kind of plants are called true breeding plants. It's possible sometimes that you have a violet color flower producing plant, you allow it to self-pollinate and it ends up producing some plants that produce white flowers. That is also possible. But such plants are not called true breeding plants. To call it a true breeding plant or a pure breeding type, that plant, if it undergoes self-pollination, it should ensure to produce all of the offspring which are similar to the parent plant. It should not show any other trait in the next generation. Okay. So how do they ensure that they have a pure breeding type of plant? They perform repeated self-pollination. Okay. See, a true breeding plant is there. I'm sorry, a tall plant is there. This tall plant is undergoing self-pollination one time, second generation, third generation, fourth generation, successively for 4 to 6 generations. If it's repeatedly undergoing self-pollination, then obviously the offspring that it's going to produce eventually after the 5th or 6th generation will all be tall. When does a tall plant have the ability to produce a dwarf offspring? Only when a dwarf plants, it has been cross-pollinated by the dwarf plant at some point in its previous generations, only then it is possible, right? But you're making sure that cross-pollination does not happen. Other, other traits are not even, coming in contact with this tall plant. So repeatedly, if you're allowing it to undergo self-pollination multiple times, all that plant has is is the gene for tallness. If it's not undergoing cross-pollination, whatever it has, that only can get passed on to the next generation. It cannot pass on a character that it does not have, right? So we are going to ensure by repeated self-pollination that you obtain a plant which is true to its type. When it undergoes self-pollination next, it's going to produce 100% of the plants which are tall in nature or produces violet flowers in nature. So such in that way, he selected plants that were true breeding type. Okay. So for all the 14 varieties that he had, like these are 7 characters, these are the 14 traits. For all of these 14 varieties of pea plants that he had, he made sure that they were all of true breeding type by repeatedly allowing them to undergo self-pollination for several generations. So that's the first step. Once he has these true breeding varieties ready, 14 true breeding varieties ready, he allows them to undergo cross pollination. You have one true breeding tall plant, one true breeding dwarf plant, allow them to cross pollinate or perform cross pollination between them. You have one plant producing violet flowers, true breeding, another plant producing white flowers, true breeding. You perform cross pollination between them. You have one plant that's producing green color seeds, another plant producing yellow color seeds. You allow them, not allow them, you perform cross pollination between the two. So, this is the next step that he did. Like I mentioned earlier, pea plants produce flowers that are bisexual. It has both the androecium and gynoecium, and you don't disturb it, leave it to itself. It will undergo self-pollination in most cases. But right now, we want them to get cross-pollinated because we want to study how those characters get passed on. We are going to perform artificial hybridization, where before the flower, blooms or opens, even before the anther matures, you remove the stamens by emasculating it. Then you dust the stigma from the other plant onto this. Like over here, I mentioned how alternating traits, contrasting traits, two plants of contrasting traits are taken. You have one violet color flower producing plant, another white color flower producing plant. So here, this plant is considered designated as female, this is designated as male. Both of them are bisexual. If this is designated as female, then you have to cut and remove the male parts from here by a process called emasculation even before the anther matures, so that you make sure that the pollen from the same plant does not fall on top of the stigma. Then what do? We dust the pollen from the desired plant on top of it and then cover it with a bag. And within this, the fruit and the seeds will form. That will be our next generation. Those seeds you will allow them to germinate, and they will produce their own flowers and everything, and that is when you test for which character has gotten passed on. uh, are all of them of the same type, or is there a mixture of both the parents? All of those studies you can do in this generation. This generation is called the F1 generation. For F1 stands for first filial generation. Okay, so now this is the next step. First, he selected parents, pure breeding types. He performed artificial hybridization across, and following that, whatever offspring he got, he planted them, and then he got new plants, that is called the F1 generation. In the next step, he will allow this F1 generation plants to undergo self-pollination. Will it undergo self-pollination? Whatever offspring you get following that, post that, is called the F2 generation. So he will look at the ratios of appearance of violet color or white color, tall or dwarf, in both F1 generation and F2 generation, and based on his observations, he will come up with certain inferences. He will infer certain things and propose that. Okay, what exactly he does, we'll see now. He first performed what is called the monohybrid cross. Okay, mono means one, hybrid means two different varieties of plants he's combining together in the offspring, that is called the hybrid. Of course, pea plants have many characters, of which he selected 7 characters and all of that. But in a monohybrid cross, he specifically zeroes down on one character and he's focused on that character. For example, if he considers the character height of the plant, it can either be tall or it can be short or dwarf, right? So this is the one character that we are fixed on for this particular cross. Of course, there will be seed color and flower position, flower color, and all of those other characters also. But we do not worry about that as of now. We do not worry about whether those are contrasting or not. Our focus in this experiment is going to be on one character, and that's why it's called the monohybrid cross or one gene inheritance. So he chose the character height of the pea plant first. Right now, I will not talk about the phenotype or the genotype. Let's just see what results Mendel got, what is visible to the eye. We'll not talk about what happened inside the plant, okay? So what is visible to the eye is selection of parents. He first selected parents that had two contrasting traits like I told you. One is tall, the other one is dwarf. What do? They perform artificial hybridization between these two, and from this cross you obtain what is called the F1 generation. So he had one tall plant crossed with one dwarf plant. Both of these are true breeding varieties, which means they are homozygous. More about that I'll tell you later, which means they have been self-pollinated for many generations. And now these two, when they crossed, in the next generation of the progeny, all of them will appear like one of the parents and none of them will be dwarf. So only one of the two traits will be visible in the F1 generation. What is the next step in the experiment? He takes the F2, F1 generation plants and allows them to undergo self-pollination. When it's undergoing self-pollination, when this plant itself is the male, this plant itself is the female, then both the plants are considered to be tall. Both the parents are considered to be tall. So it's like a tall is getting self-pollinated. So male, female, both of them will have genes for tallness, right? So that's how it is. So when he performs self-pollination, when he allows the F1 generation plants to undergo self-pollination and gets F2 generation, he will be super surprised to look at a dwarf plant in the F2 generation. He will see some tall plants and some dwarf plants in the F2 generation, but the tall and the dwarf plants will be in a specific ratio. Among all of the plants that he obtains, 75% of them will be tall like the parent and 25% will be dwarf. So think about it. The parents themselves are not dwarf. In the first generation, there was a dwarf plant. In the next generation, second generation, no dwarf. Third generation, this dwarf character is reappearing. It's almost like this plant is like its grandparent, not like its parent, because its parents are tall. And so this was his observation after performing the monohybrid cross, and he was like really surprised by the fact that no blending happened. In the F1 generation, only one of the two parental traits was expressed, right? And in the F2 generation, both the traits were expressed specifically in 3:1 ratio. 75% of them were tall and 1%, 25% was dwarf. Every time he performed this experiment, he got the same ratio. And ultimately, the traits did not show any blending either in F1 generation or F2 generation. See, you had a tall plant and you had a dwarf plant. You did not get plants that were intermediate height either in F1 generation or in F2 generation, so it did not show any blending. So this is the observation that he could see from the experiments. Okay, based on his observations, he will infer something. What is the inference we will see now? Firstly, Mendel said, because he did not see any blending, he proposed that within the parent plants, there are discrete units that were stably being passed down. That means distinct units are there in the parents. They are not changing. However, it is present in the parent in the exact same form, unchanged, not overlapping, exactly as it is in the parent, it is getting passed on to the next generation. How is it getting passed on? It's getting passed on through the gametes. That was his first inference. Whatever those units are, whatever is getting passed on from one generation to the next generation via gametes, he gives them a name. He calls them factors. Okay. Whatever Mendel referred to as factors, today we call them as genes. Today we know that genes are the units of inheritance. When reproduction happens through the gametes, it is our genes that are getting passed on from one generation to the next generation. So if inheritance of characters are happening, it is happening through the genes. That's why we call genes as the unit of inheritance as well, right? So the genes is what contains information that is required for the expression of character in any organism. I am passing on my black hair color to my son through my genes. In my body, there are cells which have genes responsible for black color. Those genes I'm passing on through my gametes to my son, so my son ends up having that character, right? So it is through the genes that inheritance happens. That's why they're called the units of inheritance. Now, in different organisms, genes can vary, okay? For example, we know that gene is nothing but DNA, right? So, genes are DNA. Within DNA, you have specific sequences of nucleotides A, T, C, G, C, G, T, A, whatever. Okay, so I'm writing a random sequence. Of course, the gene is not going to be so short. It's going to have at least a few hundreds to thousands of nucleotides. Just for our reference, I'm showing you this. Okay, so this is how a gene will be. Okay, so let's say this gene is responsible for eye color. Okay, this gene is responsible for eye color. All humans will have a gene responsible for eye color. But in me, there is dark brown eye color. The color of my iris is dark brown. Most Indians, it's dark brown. And in some of them, it could be blue. In some of them, it could be green. All of them have a gene for this particular character. Okay, it's present in the same location on our chromosomes also. For example, if this is the chromosome, at this location on the chromosome, this gene will be present. If this is chromosome number X, let's say, okay. So in all individuals, if it's chromosome number 1, in every individual chromosome number 1 at this position, there will be information for hair color, eye color, like that, okay. So now we are taking the example of eye color, let's stick to that. So at this position, in chromosome number X, it could be 1, 2, 3, whichever, I'm not sure which chromosome has the information for eye color. Let's just take X for example. And if in this chromosome there is information for eye color in me, then in all humans in that particular chromosome at that particular location there will be information for eye color itself. But what is present in the gene can vary. If the gene in this position is GC, then probably the eye color of that individual would be blue. If in that gene instead of GC there is CG, that could give rise to green eye color. If in that region there is information for AT, that could give rise to brown eye color. Okay. So this gene can exist in different forms. If this is the sequence, it's giving rise to blue. If there's any change in the sequence, it could be some other color. If there's some other change, that could be some other color. So this single gene can exist in different variants. The different variants of a gene, the different forms of a gene is what we refer to as alleles. Okay. So the character that we are considering here is eye color. For eye color, there is one gene responsible. This gene is present in the same location in all individuals on that same chromosome itself. But the sequence of nucleotides in this gene can vary. If the sequence varies, this gene, whatever variants you have of this gene, they are called alleles. Over here, there are three different types of alleles, allele 1, allele 2, allele 3. Because in the sequence, there are three different variants. Okay. So, this allele 1 will give rise to, let's say, brown eye color. Allele 2 gives rise to green eye color. Allele 3 gives rise to blue eye color. These three variants of this character are called traits. Eye color is the character, brown, green, blue are the traits. Genes are responsible for eye color, but the different variants of the gene are what we call alleles. So brown eye color is because of this allele. Green eye color is because of allele 2. Blue eye color is because of allele 3. What are these alleles? Alleles are different forms of the same gene. How does one allele differ from another? With respect to its nucleotide sequence. Okay. So, he says that, today we know that genes which code for a pair of contrasting traits are known as alleles. They are slightly different forms of the same gene. How are they different? They are different with respect to the nucleotide sequence. Alright, now let's move on. So, how did he infer that? He studied, he performed his experiment, he looked at the results here based on his observation. He inferred all of those 4 points that I told you. Okay. Now, how exactly did he come to that conclusion? How did he infer that? He said that every character in a plant is controlled by a pair of factors. Okay. Now, this is a tall plant. Within the cells of this tall plant, inside the tall plant cells, there are those factors, right, that are getting passed on from one generation to another. The tall plant has factors responsible for tallness. The dwarf plant has factors responsible for dwarfness. That's dwarf factor, it is getting passing on to the next generation. The tall factor, this will pass on to the successive generation. He represents that using letters, alphabets. Okay. Now, whichever plant was observed in the F1 generation, see, when he performed the experiment, he saw how whenever tall and dwarf plants were, crossed, he got all tall plants. Whichever plant, whichever variant, whichever trait he observed in the F1 generation, he calls that the dominant trait. Which trait does not appear in the F1 generation, that is called the recessive trait. Okay. So over here in this experiment, tallness is dominant, dwarfness is recessive. Okay. So we know that he inferred that there are factors. Each character is controlled by a pair of factors. So we know that since pea plants are diploid like us, for each character there will be two copies, one that it got from the mother, one that it got from the father. So this is a pure breeding tall plant. If it is going to produce all tall plants, if it undergoes self-pollination, then all it has is information for tallness, right? What we know is that each plant has two copies of information for each character. What we know is the parents that we selected were true breeding types. So, if it is true breeding type, that means it should have information only for that particular trait. This dwarf plant has information only for dwarfness. The tall plant has information only for tallness. So, each of them have two copies of information for height, but both of them are identical. This has both tall information factors, this has both dwarf information factors, okay. So here we are talking about the height. It has two variants, tall and dwarf. Since tall is the dominant for representation purpose, we are going to use capital T as the allele for tallness, lowercase t for the allele for dwarfness, okay. Now we have to understand two terms, phenotype and genotype. When you look at a plant that is tall, that is its phenotype. You look at it physically, you can observe it. You don't have to cut through the plant or anything. It's easily visible. The external features, that is the phenotype. Genotype is the genes that is responsible for that phenotype, which you cannot see. That is present inside the cell within the DNA. That is something that you cannot see. We represent that using letters. Okay. So here, this is a true breeding pea plant whose phenotype is tall. Its genotype is capital T because it is a true breeding variety. Next, you have a pea plant that is dwarf, true breeding variety. Its genotype will be lowercase t. It will have only alleles for dwarfness. Now, let's see what happens in the cross by taking both the phenotype and genotype into consideration. Let's start off with the parental generation. What is the phenotype in the parental generation? We have one tall plant that is crossed with a dwarf plant. What is the genotype? This has both tall alleles. This has both alleles for dwarfness. How are the gametes produced here? We know that whenever sexual reproduction happens in diploid organisms, gametes will be produced haploid, right? Haploid gametes are produced. So when this plant undergoes sexual reproduction, it produces gametes. The gametes will have only one of the two alleles. Since both the alleles are of the same type, all of the gametes will have capital T allele within it. And here in this case, all of the alleles will have, all of the gametes will have lowercase t in it. So, when these two come together in a hybrid in the F1 generation, we saw that the phenotype was tall. What would the genotype be here? The genotype here would be capital T lowercase t. I want you to notice how even when the genotype is capital T, the phenotype is tall. Even when the genotype is capital T lowercase t, phenotype is tall. Even when lowercase t, that is responsible for dwarfness, is present in this plant, it is still displaying only tallness. Okay. So, keep this in mind. What are we going to do next? You will take the F1 generation plant and self it, allow it to undergo self-pollination. Selfing F1 hybrid. What would the phenotype be? The phenotype would be tall crossed with tall. What would the genotype be in this case? We are talking about the F1 hybrid. All of them are tall and the genotype is capital T lowercase t crossed with capital T lowercase t. What kind of gametes will get produced here? Since you have two different types of alleles in this particular plant that we are considering right now, 50% of the gametes will have capital T, 50% of the gametes will have lowercase t. Okay. So here each plant, male parent, female parent, both will produce two types of gametes. Let's see what are the different possibilities we get in the F2 generation by using what is called a Punnett square. Punnett square was developed by a British geneticist whose name is Reginald Punnett. Okay. So whenever you're drawing a Punnett square, you have to check how many gametes are there in each parent. Right now it's 2 and 2. So you will draw a square that has 3 rows plus 1. Here you have 2, so plus 1 you do. Your Punnett square should have 3 rows and 3 columns. I have drawn 3 columns now. Let me draw 3 rows. 1, 2 and 3. In the first row, we will take the gametes. This side you have the female gamete, here you have the male gamete, here you have the female gamete. The gametes occupy the first row and the first column. So you have capital T, lowercase t here, capital T, lowercase t. This will be capital T, capital T, capital T, lowercase t, capital T, lowercase t, lowercase t, lowercase t. So we know that whenever you have capital T in the genotype, the plant will definitely be tall. Whenever you have both lowercase t, then the plant is going to be dwarf. If you look at the F2 generation, you will see there are two different types of phenotype. Either they are tall or they are dwarf. So you get the phenotypic ratio 3 tall is to 1 dwarf. Look at how many genotypes are present. You have capital T, one type, capital T, lowercase t, second type, lowercase t, third type. There are 3 different types of genotype. That is the genotypic ratio. How many capital T do we have out of 4? We have 1 capital T, 2 capital T, lowercase t and 1 lowercase t. That is the genotypic ratio. See, when I told you initially that Mendel chose pea plants that were pure breeding, true breeding type, this is what I meant. If he considers this kind of tall plant as the parent, then in the offspring, you can end up getting some dwarf plants also. So this is not true to its trait. It can show another trait. Like this plant by itself is not tall. Is not dwarf. It is showing dominant phenotype itself. But its offspring can be tall. So this is not called true breeding pea plant variety. This is a true breeding pea plant variety because if it is self-pollinated, it will produce only tall. Why is this going to produce tall all the time? Because all it has is alleles for tallness. Why is this producing dwarf sometimes? Because it has a dwarf allele also. It may not be expressing it, but it has that allele. So that's why it is possible for it to produce offspring that are dwarf. Okay. So now there is a term that we need to understand. Every time you take a character, for example, height of the stem in pea plant, let's consider this. There are two possibilities. Either it can be tall or it can be dwarf. A tall plant appears in the F1 generation, so that is said to be dominant, and the one that does not appear in the F1 generation is said to be recessive. Now, the tall plant can exist in two forms, capital T or capital T lowercase t. The dwarf plant will always exist in lowercase t lowercase. Whenever the tall plant has both alleles that are similar, that are the same, then you call it homozygous. In the zygote, similar alleles are coming together. This is homozygous. If you have two different alleles present in the hybrid, then you call it heterozygous. So a tall pea plant, a pea plant that is displaying a dominant phenotype, its genotype can either be homozygous or it can be heterozygous. But for a plant that is displaying the recessive phenotype, it is always 100% of the time always homozygous. This can never be heterozygous. All the plants that are showing the recessive phenotype, they will always be true breeding types only. Tall plant showing the dominant phenotype, you're not sure. By looking at it, you can't say whether it is a true breeding type, whether it is dominant or recessive. Okay. So when Mendel selected the parents for experiment, this is what I was talking about. He was looking for homozygous plants. Then if I had told you homozygous, you would not have been able to understand. Now I hope it makes sense to you. Okay. Yeah. So is there any way to check whether the tall plant is homozygous dominant or heterozygous dominant? There is a way, and that is called the test cross. What we do in a test cross is, what do we do in a test cross? You take the plant that is showing the dominant phenotype. So you're taking a tall plant. Now we do not know whether this tall plant is capital T or capital T lowercase t. It could be either. Okay, so this tall plant can either be capital T or it can have the genotype capital T lowercase t. Regardless, we will cross this with a plant that is showing the recessive phenotype. Based on the results you get from this cross, you can identify whether this pea plant was homozygous or heterozygous. If it is homozygous, then all of the offspring will be tall. If it was heterozygous, then you will get 50% tall plants, 50% dwarf plants. So you will get offspring in 1:1 ratio. If you get offspring in 1:1 ratio where 50% are tall, 50% are dwarf, then that means this plant is heterozygous. If you get all tall plants, that means that plant is homozygous. So the cross that we perform to check whether the plant showing the dominant phenotype is homozygous or heterozygous, this is the cross we perform. This is called the test cross. Again, based on the test cross results, you can determine whether the plant was homozygous or heterozygous. That is not the case for plants that are showing recessive phenotype because they cannot be heterozygous ever. They will always be homozygous. So that's the case. Yeah. So based on his monohybrid cross experiments, Mendel proposes two laws. The first law is called the law of dominance and the second law is called the law of segregation. First, let's see what the first law of dominance states. It has 3 points. The first point is that characters, all the characters in living organisms, it could be eye color, hair color, leaf position, flower color, all of these, they are controlled by units, and these units are discrete. Okay, discrete means they are distinct, they don't overlap. Or they do not blend, we can write. So he says that whatever characters, whatever units are controlling factors, they're very discrete. They do not mix up in between. They do not change how it was in the parent exactly like that. It is going to get passed on to the offspring. He gives it a name. He calls it factors. And these factors, he says, always occur in pairs. Of course, because we are diploid organisms, we get one set from the male parent, one set from the female parent. So it exists in two forms. Now, whenever it exists in pairs. Now, whenever in that combination, you have a pair of dissimilar alleles. Dissimilar means heterozygous condition. Okay, so whenever there is dissimilar pair of factors, which means heterozygous, like in the case of capital T, lowercase t, whenever it is in dissimilar pair like this, not capital T or lowercase t, whenever it is in heterozygous condition like this, one member will dominate and express itself. Whenever this is the combination, the plant will be tall. So tallness allele is expressing itself while dwarfness allele is not expressing itself. Whichever one expresses is called the dominant allele. The one that doesn't express itself is called the recessive allele. That is the first law of dominance. The law of dominance, which is called the first law of inheritance. He performs experiments with all the characters that he chose for pea plant, just like the height of the stem. For each one of them, he will identify which is the dominant trait, which is the recessive trait. You must know which is dominant, which is recessive. It's very important. So for height, we already know tall is dominant, dwarf is recessive. For flower color, violet is dominant, white is recessive. For the flower's position, axial, at the axil of the leaves is dominant. Terminal, at the tip of the plant is recessive. For the color of the pod, the fruit color, green is dominant, yellow is recessive. For the fruit shape, inflated is dominant, constricted is recessive. For the seed color, yellow is dominant, green is recessive. For the seed shape, round is dominant, wrinkled is recessive. Here, pod color, seed color, both are having the same color variants. Pod also has green and yellow, seed also has green and yellow. But notice the difference in which is the dominant trait. In the pod, green is dominant. In the seed, yellow is dominant color. You tend to think that the peas that we eat are usually green in color, so probably that's the dominant one. That is the predominant one that is mostly found in the population. But what dominant means is whenever it is in heterozygous condition, whichever one expresses, that is called the dominant one. So in the presence of the green color allele, the yellow color allele will express itself. So that's why yellow is dominant. Okay, so keep that in mind. Don't get confused with between these two. Both of them have green and yellow variants, but in pod, green is dominant. In seed, yellow color is dominant. Now, what is the second law of inheritance? It is called the law of segregation. So he notices that there is no blending that is seen either in the F1 generation or the F2 generation for all of the 7 characters that he considered. He proposes this law. He says even though the parents have a pair of alleles or a pair of factors, the parents contain 2 alleles. During gamete formation, our cells will have 2 alleles because our germ cells that form the gametes are diploid. There will be 2 alleles. But during gamete formation, what happens is these 2 alleles or factors segregate in such a way that the gametes will receive only one factor. This he is told at a time when he did not even know about what meiosis is, what happens during cell division, what happens during gamete formation. He had no idea about that. But he said, even though we have 2 copies of alleles, even though our factors are paired, during gamete formation, these 2 will segregate in such a way that the gamete will receive only either one of them. Only one of them will be received. So it will get segregated during gamete formation, he said, even without having an idea about meiosis. So this is very significant, and this is true for all diploid organisms. In all diploid organisms, be it plants, animals, you will notice how during gamete formation, only one set of alleles or one set of chromosomes get passed on to the gametes, right? So this is called the law of segregation. Now, similar to the experiments that Mendel performed, many other scientists tried to replicate the same experiments using some other organisms. There was a group of scientists who performed monohybrid cross experiments on a plant that is called snapdragon or antirrhinum or dog flower. All of them refer to the same plant. And the character that they chose was the color of the flower. The color of the flower was either red or white or pink in a population. Okay. So in the first generation, they chose plants that were red, that produced red color flowers, and another set of plants that produced white color flowers. In the parental generation, red and white were chosen. When they were crossed, the steps that they performed were very similar to the Mendel's monohybrid cross experiment itself. They take the parental generation that are true breeding, and then they will perform artificial hybridization, cross pollination. Following that, what you get is the F1 generation. On selfing the F1 generation, you will get F2 generation. That's the same steps that they followed too. But to their surprise, in the F1 generation, they obtained pink color flowers. Now, there are some things that we must understand here. In the parental generation, you have red flowers, you're crossing them with white flowers. According to Mendel's law of dominance, either the F1 generation should have all red flowers or it should have all white flowers. If it shows all red flowers, that means red is the dominant allele. If it shows all white flowers, that means white is the dominant allele. But look at this. It's neither red nor white, but it is a brand new phenotype. The parents were, neither of them were pink. But in the offspring, you're finding pink color producing flowers. So it's a new phenotype that is seen in the F1 generation firstly. And this new phenotype is not a random color. It's not like red and white color produced yellow color flowers, red and white color produced orange color flowers. They've produced a color which is intermediate between red and white. You mix red and white paint, you get pink color, right? So this is intermediate between red and white. They got a brand new phenotype which was not present in either of the parents, and that was an intermediate phenotype. Okay, so in the F1 generation, after they allowed it to undergo self-pollination, in the F2 generation, they noticed the appearance of red, white and pink in 1:2:1 ratio. 25% of them were red, 50% of them, this is not 75, this is 50% of them were pink, and the remaining 25% were white. Okay, so this kind of inheritance is called incomplete dominance. This is the actual flower, antirrhinum flower. This is the red variant, this is the white variant. When they are crossed, you get a pink variant. Okay. So what exactly happens here? Let's try and understand. We'll start with the parental generation. In the parental generation, what is the phenotype? What is the genotype? What are the gametes? We will write down. Here, the crossing, red flower with white flower. This would be the gamete. When these two gametes come together in the F1 generation, the phenotype is brand new, the genotype is heterozygous. The phenotype will be pink and the genotype will be heterozygous, right? Now, when we allow this heterozygous plant, you can't call them dominant now because it's not completely dominant. If you perform self-pollination to the F1 hybrid, write down the phenotypes and genotypes. You have pink flower, crossed with pink flower, capital R, lowercase r, crossed with capital R, lowercase r. Now it will produce two different types of gametes because they are heterozygous. 50% of the gametes will have capital R allele and the other 50% of the gametes will have lowercase r allele. Capital R allele is responsible for red color, lowercase r allele is responsible for white color. So what do we get? We get F2 generation. Let's draw a Punnett square. In the F2 generation, since there are two gametes each, we will draw a Punnett square that has 3 rows and 3 columns. You have capital R, lowercase r, then you get these possibilities: capital R, capital R, capital R, lowercase r, capital R, lowercase r, lowercase r, lowercase r. Whenever you have both dominant alleles, it will produce flowers that are red. Whenever you have a heterozygous condition with one dominant and one recessive alleles, the flowers are going to be pink. When you have two recessive alleles coming together, then the flower is going to be white in color. So in the F2 generation, the phenotypic ratio is going to be 1 red, 2 pink, 1 white. And the genotypic ratio would also be 1 capital R, 2 capital R, lowercase r, and 1 lowercase r, lowercase. Okay. Here, if you compare this with Mendel's monohybrid cross that we studied about height of the plant, you will notice how in this case, the phenotypic ratio and the genotypic ratio are the same. Whereas in the monohybrid cross that we performed here, the phenotypic ratio was 3:1 and the genotypic ratio was 1:2:1. Here, the phenotypic ratio is there. In incomplete dominance, you got 1:2:1 because there are 3 different phenotypes there in the F2 generation. Here you have only 2 phenotypes, either it is tall or it is dwarf. But over there, you have 3 different phenotypes. Okay. So this is called incomplete dominance. Whenever it is in heterozygous condition, it will produce a brand new phenotype which is not there in either of the parents, and that phenotype will be blending. So this capital R allele does not completely dominate the lowercase r allele. Whenever it is in heterozygous condition, it only partially expresses itself. So you get an intermediate phenotype. Now if you look at this incomplete dominance and think about the law of dominance, we can say that the law of dominance is not true for all organisms and all characters. It was true for the pea plant. It was true for the characters that Mendel specifically chose in the pea plant. We have replicated the exact same experiment in another organism for another character. Over there, this did not hold true, right? What does Mendel say? Whenever there is a pair of dissimilar factors, one will express, one will not express. Whichever expresses is dominant, which does not express is recessive. But did that happen here? That did not happen here. In the heterozygous condition, according to Mendel's law of dominance, R should have expressed, all of these should have been red, and lowercase r should not have expressed, or lowercase r had expressed and white color would have expressed and red color should not have expressed. Whichever one was dominant, that should have expressed, but that did not happen here. So this incomplete dominance is a deviation from Mendel's law of dominance. Mendel's law of dominance does not hold true here. Mendel's law of dominance is not universal. It does not apply to all organisms for all characters. It is only true for those characters in pea plants which Mendel chose. Not everywhere. It's not universally applicable. So, this is not a universal law. This incomplete dominance is said to be a deviation from Mendel's law of dominance. What about law of segregation then? Law of segregation is universal. It applies to all organisms that are diploid. In all diploid organisms, there are one pair of alleles for each character. And during gamete formation, only one of them will get passed on to the next generation. Okay? Now, let's understand the concept of dominance. Now, what exactly is dominance? We saw how in Mendel's monohybrid cross, when there was a pair of dissimilar alleles, one of them expressed itself, we called it the dominant. However, that did not seem to happen in snapdragon flower color. There, it exhibited incomplete dominance. So, what do we mean when we say an allele is dominant over the other? What is happening at the genetic level? What determines dominance? We'll try and understand that. What we already know now is in our chromosomes, there are many genes present. All of us have two sets of chromosomes, all diploid organisms. One we receive from the mother, one from the father. Within these chromosomes, there will be many genes, and genes are nothing but sequence of nucleotides. Now, in an individual, there might be similar type of alleles from both parents. Like for example, if both your parents have black color hair, you will have alleles for black color hair, right? So that in that case, when both the alleles are same, then we call it homozygous. It's possible that for a particular character, we might have two different types of alleles. If the father has black hair and the mother is blonde, then you will have two different alleles, in which case we will call it heterozygous. Okay. In all diploid organisms, genes can be homozygous or they can be heterozygous. Now, in a population of humans, today we see so much variation, right? If a hair color can exist in so many different forms, that means the gene responsible for production of hair color has so many different variants, it has so many different alleles. How did so many alleles arise in a population? We'll understand that. Now, when initially human population evolved on this planet, it's possible that all of them had similar kind of alleles, all of them probably had black hair. Now, in DNA, there are changes that take place which we call mutation. There will be changes in the nucleotides that take place which we call mutation. Sometimes mutations may have happened in those genes because of which the sequence of nucleotides have changed. Once that sequence of nucleotide has changed, it has no longer been able to function the same way it did. Because of that modification in nucleotide sequences, because of mutation, newer alleles may have formed in the population. Initially, there was just one wild type, one normal allele which all individuals had. So, for example, this is the allele that everybody had, let's say. In this position, one person got a mutation. Instead of CG, there is AT right now. Because of this one mutation, it's possible that this allele will no longer work like this. It will show a new phenotype. It's also possible that many other individuals underwent similar mutations, like in different parts of the DNA segment, and they have given rise to newer alleles. Okay, so this is modified because of mutation. Newer alleles that form are modified because of mutation. Now, a normal allele, let's say it's supposed to produce uh, a pigment responsible for hair color. Okay, so the normal allele is going to produce an enzyme which is responsible for the formation of pigment. You know, in our bodies and living organisms, for any reaction to take place, enzymes have to be there. They will catalyze the reactions. For the formation of this pigment also, one enzyme is essential. And that enzyme is produced, the information for that enzyme is present in this location in this particular gene. Okay. So this gene produces that enzyme. Because of that enzyme, we are going to get the pigment which is responsible for black hair color. Now, this gene is mutated. This allele is mutated, it is modified. Now, this modified allele, what is going to happen with the modified allele? One of three things can happen. Either the modified allele is just changed a little bit compared to the normal allele. It is able to function as the normal allele itself, but the enzyme that is produced may be slightly less efficient. So, the products are formed, but the concentration of the product might be little less, or the allele might not be affected at all. It might function as the normal allele itself. Okay. Secondly, the allele is modified in such a way that the enzyme that is produced varies in shape. You know how enzymes and substrates function, right? The active site of the enzyme has to be specific. The substrate can come and bind to that site only after the binding and enzyme substrate formation will the bonds form or break, and eventually products are formed. If the shape of the enzyme is altered, the enzyme will not be able to function. So with the second type of modification, what could happen is the enzyme that is formed is not able to do its job. It is not functional. It is formed, but it's not able to do its job because it is modified. If it is not able to do its job, even though the enzyme is produced, the products will not form. So that pigment will not form. Instead of that, some other pigment will, some other color can appear. A lighter color can appear because the dark pigment is not forming. Let's say the allele is modified in such a way that the enzyme itself is not produced. If the enzyme itself is not produced, then obviously there won't be any products. So because of such modifications in alleles, you can see that sometimes the phenotype is expressed, sometimes it's not expressed. Let's take the example of height in pea plant. Capital T allele expresses tallness, lowercase t is dwarfness. What does this mean? The gene responsible for the height has two variants. One is capital T, one is lowercase t. Capital T is actually responsible for the production of an enzyme involved in gibberellic acid synthesis, gibberellin synthesis. You know, gibberellins help in internodal elongation in plants, right? So when gibberellins are synthesized, internodal elongation happens and the plant grows tall. If lowercase allele T is present, then that allele is modified in such a way that the enzyme required for gibberellin synthesis is not produced. As a result, gibberellins are not produced, the plant will not grow tall. Got my point? So that's how recessive alleles are formed. Usually the wild one, the one that has been present since the beginning, that will be the dominant one. The ones that are modified later will usually be the recessive alleles. Okay. So that's about what is the concept of dominance. Now let's learn about one other deviation from Mendel's law of dominance, which is called codominance. Over here, we are taking the example of blood groups. You know, there are 4 different phenotypes for blood groups. Blood group can be A, B, AB or O. Now, what determines the blood group of a person is the kind of antigens that are present on the surface of our RBCs. RBCs are red blood cells, right? So, these RBCs are sugars that are located on this plasma membrane, within the plasma membrane of RBC. These antigens are of 2 types, either A antigen or B antigen. If RBC surface has only A antigens, you refer to it as A blood group. If it has only B antigen, then you call it B blood group. If they have both A and B, then you call it AB blood group. If they have neither A nor B, then you call it O blood group. Okay. So, these are the 4 different types of blood groups. The character blood group is controlled by one gene that is called I gene. The I gene has 3 variants, meaning it has 3 alleles. It can either be I superscript A, I superscript B or lowercase I. If it has I superscript A, it's going to produce the A antigen. If it has I superscript B, it's going to produce B antigen. If it has lowercase I, it's going to produce no antigen. Okay. So, if a person should be of O blood group, then they shouldn't have either of these. If these are there, A or B antigens will be produced. So, an O blood group person will have both alleles that are lowercase I, lowercase I. A person having A blood group should have at least one copy of this allele. It can have both copies of this or at least one copy of this. So, A blood group can either be homozygous dominant like this or it can be heterozygous. Same goes for B blood group. If a person should have B blood group, they should have at least one copy of I superscript B. They can have both or at least one. They can be homozygous or heterozygous. Now, speaking of AB blood group, on the RBCs, you have both A and B antigens present. That means in them, one copy of both these must be present. So, AB blood group will have this genotype IA, IB. Over here, you have 4 phenotypes O, A, B, AB and 6 genotypes. O and AB have one genotype each. O is always homozygous, AB is always heterozygous, but A and B have 2 different genotypes each. So they can be homozygous or heterozygous. So there's a total of 6 genotypes for 4 phenotypes. Over here, codominance is observed only in the AB blood group. Co-dominance. Co means what? Co means together. When both the alleles are expressing simultaneously, then you call it co-dominance. According to what Mendel said, when there is a pair of dissimilar alleles, one will express, one will not express. The one that expresses is called dominant, right? That is Mendel's law of dominance. But over here, that is not applicable because over here you're looking at a heterozygous genotype. They are a pair of dissimilar alleles, but not only one of them is expressing, both of them are simultaneously expressing. That's why we call it codominance. Now, when we speak of an example for codominance, we don't say ABO blood group is an example for codominance. Codominance is seen specifically in AB blood group. Look at A and B blood groups. Here you have heterozygous condition. Here also you have heterozygous condition. Over here, in the heterozygous condition, it is showing complete dominance similar to Mendel. Only A is expressing, B lowercase I isn't. Only IB is expressing here, lowercase I isn't. So when you consider these three alleles, it's like IA and IB are equally dominant, okay? But lowercase I is recessive with respect to IB as well as IA. So in this combination, it's showing complete dominance. IA and IB is showing codominance. Okay, so only AB blood group is example for codominance. All other blood groups will show complete dominance, similar to Mendel's dominance. Okay, so this codominance again is a deviation from Mendel's law of dominance. Now, these are the three different patterns of inheritance that we've learnt about so far. We spoke about dominance in Mendel's monohybrid cross, where the offspring will resemble one of the two parents. In incomplete dominance, the F1 generation hybrid will resemble neither of the parents. It will have a brand new phenotype which is intermediate to both the parents. In codominance, you will see that the offspring will express both the traits simultaneously. For example, if you have a brown colored cow and a white colored bull, and if they are mating, their offspring will show patches of brown and white color coat on its surface. So it's expressing both the traits simultaneously. That is called codominance. AB blood group again is an example anyway. Now, ABO blood group is also an example for multiple alleles. Multiple means what? Multiple means many. For a particular character, if there is more than two alleles, always when we say many, we say more than one, but keep it in mind, whenever there is more than two alleles for a particular character, for a particular gene, then we call it multiple alleles. We saw more than two is 3, minimum is 3. We saw how the gene that is responsible for controlling blood groups has 3 alleles, IA, IB and lowercase I. So that is an example for multiple alleles. Okay, so ABO blood group is example for multiple alleles, but AB blood group specifically is an example for codominance. So sometimes it so happens that one gene will have more than one phenotypic effect. Okay, so for example, a single gene can have more than one effect. This can be seen in pea plant again. So in the pea plant seeds, in the pea seeds, there is one gene that is responsible for the synthesis of starch grains. Inside the pea seed, there will be starch grain synthesized, right? So the starch grain synthesis is controlled by single gene. This has two variants. It can either be capital B or it can be lowercase p. If it has two variants, it can exist as 3 genotypes, right? It can be capital B, it can be capital B, lowercase p, it can be lowercase p, lowercase p. Now, this one gene controls two characters. One thing is the size of the starch grains, and the other thing is the shape of the seed. Both of these characters is controlled by this one gene itself. So basically, this gene is going to produce an enzyme that is involved in starch synthesis. When both of the alleles are dominant, capital B, then starch synthesis will happen very, very efficiently. As a result, the size of the starch grains, here I'm not talking about the size of the seed, I'm talking about the size of the starch grains within the seed. The size of the starch grains is going to be large. Whenever the size of the starch grains is going to be large, the shape of the seed is going to be round. Okay, so the seed is going to be like this. If this is the seed, it's going to be round, and inside that the starch grains are large. If it is in heterozygous condition, efficiency is slightly reduced. The size of the starch grains is intermediate, not as large as it was in homozygous dominant condition. So here the shape of the seed is round itself, but the size of the starch grains is slightly smaller. When both of them are lowercase p, then the size of the starch grain is also small and the seed is also wrinkled. If you look at size of the starch grains, you see incomplete dominance here. When it is homozygous B, it is very large. When it is homozygous lowercase p, it is very small. In the heterozygous condition, it is showing an intermediate phenotype where the size of the starch grains is not as big as this, not as small as this, intermediate. But when you look at the shape of the seed, you have only two phenotypes. Either it can be round or it can be wrinkled. So this single gene that is controlling two effects, you can see how when you consider size of the starch grains, it is showing incomplete dominance. It's showing incomplete dominance here. Whereas when you speak of the shape of the seed, it is showing complete dominance. So what do I do now? Should I call this gene dominant or incompletely dominant? We cannot call the gene itself as dominant or incomplete dominant. We cannot, say that this allele itself is dominant or incompletely dominant. It depends on the context. It depends on which character we are examining. If you're considering the size of the starch grains, it's showing incomplete dominance. If you're considering the shape of the seed, it's showing dominance, right? So we say that dominance is not an autonomous feature of a gene. Whenever a gene is controlling more than one effect, then you cannot say for sure that a particular allele is dominant or recessive or incompletely dominant or whatever. That is that there's one statement in NCERT which says dominance is not an autonomous feature of a gene. Okay, so that is what it means. Whether that gene is dominant, that allele is dominant or recessive depends upon which character we are considering and looking at. Okay, now let's speak of the dihybrid cross. Similar to how Mendel performed a monohybrid cross, he also performed a dihybrid cross where he at a time considered two characters. Previously in monohybrid cross, we specifically studied only one character, which was height of the plant. In a dihybrid cross, he focuses on two characters at a time. We will consider one dihybrid cross, for example, which is considering two characters, and those two characters are with respect to the seed. One is the shape of the seed and the other is the color of the seed. The shape of the seed could be round or wrinkled. The color of the seed could be yellow or green. Now both of these traits, round and yellow, these are dominant. Both of these, wrinkled and green, these are recessive traits. So when he selects parents, he makes sure that one parent has both dominant alleles and the other parent is showing both recessive traits. So one of the parents will have seeds that are round and yellow, will produce seeds that are round and yellow, and the other parent will produce seeds that are wrinkled and green. Okay? So round and yellow, wrinkled and green. This is the combination of characters that Mendel selects for his dihybrid cross. Now, let's represent that. Parental generation. What is the phenotype and genotype? Phenotype is round and yellow, crossed, wrinkled and green. Genotype is capital R, capital R, capital Y, crossed with lowercase r, lowercase r, lowercase y, lowercase y. Why there are four alleles, four letters that I'm representing right now? It's not because it's a tetraploid organism, it's because we're taking two characters at a time. Each character will have two alleles, so there's a total of four alleles from two characters. Okay. So now, when these gametes form in these parent plants, each gamete will have one copy of each allele. Like from this there will be one copy, from this there will be one copy. Since both the alleles for a particular character are same, they are homozygous, we will get just one type of allele. Here we will get just one type of allele. When these two come together in a hybrid, what do we see in the F1 generation? In the F1 generation, the phenotype and the genotype will be round and yellow. It will display both traits that are dominant, and the genotype will be heterozygous for both the characters. What do we do next in the experiment? On selfing F1 generation, what do we get? The phenotype and genotype round in yellow, self-pollinated. So round in yellow, both the parents will be heterozygous for both the traits, both the characters. What kind of gametes will this produce? How many variations can you see? Now, if a genotype is given to you, if you want to see how many possible gametes can be formed, there's one formula. 2 to the power of n, where n represents the number of heterozygous genes. Over here, in this, we can see 2 heterozygous genes. So 2 to the power of 2 is equal to 4. So this can produce 4 different kinds of gametes. If you apply the same over here, there are no heterozygous genes at all. So 2 to the power of 0 is 1. Only one kind of gamete it can produce. So if you take capital R, lowercase r, capital Y, here there is only one heterozygous gene. So 2 to the power of 1 is 2. This genotype can produce 2 different types of gametes. That way you can get to know how many gametes can be obtained for a particular genotype. What are the possibilities of gametes? So here we have capital R, lowercase r, capital Y, lowercase y. How do we obtain gametes for this? Do it like A plus P multiplied with C plus D. How do you go about with this? A multiplied with C plus A multiplied with D. Then B multiplied with C. Then you do B multiplied with D. Right? Very similarly, when you have capital R, lowercase r, capital Y, lowercase y, you can't have capital R, lowercase r in a gamete or capital Y, lowercase y in a gamete. Each gamete should have one allele for both the characters. So all of the gametes should have one R, one Y. So what do we do? First capital R, capital Y, capital R, lowercase y, lowercase r, capital Y, lowercase r, lowercase y. That way we get 4 different kinds of gametes. Okay. Now, what happens here is we get 4 different kinds of gametes from each parent. Capital R, capital Y, capital R, lowercase y, lowercase r, capital Y, lowercase r, lowercase y. Same thing over here as well. When you check the F2 generation, the different combinations in the F2 generation. When we draw a Punnett square, since we have 4 gametes right now, we need a Punnett square that has 5 columns and 5 rows, right? 1, 2, 3, 4, 5. Out a little bit. Here you have 1, 3, 4 and 5. Okay. Now we'll take male gametes on one side, female gametes on the other side. So when you plot this in a Punnett square, you can fill all of the genotypes. You will get round and green, 9 of them. I'm sorry, round and yellow they are, not round and green. You get 9 round and yellow. In all of these boxes, you will get round and yellow. I'm talking of the phenotype. You can fill up the genotypes as homework. Here, in these 3 boxes, you get round and green. In these 3 boxes, you will get wrinkled and yellow. In the last box, you get wrinkled and green. What combination did we have here? We had round and yellow, round and yellow, round and yellow, round and yellow. This parent was wrinkled and green, right? If you look at the F2 generation, we get a phenotypic ratio of 9:3:3:1, where 9 are round and yellow, 3 are wrinkled and yellow, 3 are round and green, 1 is wrinkled and green. Now, if you observe these phenotypes, you will notice how this phenotype and this phenotype, both of these combinations have existed in the past. Round and yellow was there in the parental generation in the F1 generation. Wrinkled and green was there in the parental generation, right? So these are combinations that are already existed in the previous generations. So these are called parental combinations. But if you take a look, you have green that is round, which did not exist either in the F1 generation or in the parental generation. You have yellow that is wrinkled, which did not exist as a combination either in the F1 generation or in the parental generation. These combinations are new right now. Okay, so these are called recombinants. Among the 16 possibilities here, there are total of 10 parental types and 6 recombinants. How is it possible for newer combinations to form? How did these combinations that did not even exist in the previous generations form in the F2 generation? This is what Mendel was trying to understand. Imagine it like this. There's one father who has red hair that is straight, and the mother has curly hair that is blonde. So red is always with straight, curly is with blonde. If the children end up having red hair that is curly, it's a new combination. That is called a recombinant. Okay, likewise here, these two combinations did not exist earlier. Round was there, but it was yellow. Every time it was round, it was yellow. Every time it was wrinkled, it was green. But right now we have a yellow that is wrinkled and a round that is green. So how did these new combinations form? To explain that, Mendel proposes what is called the law of independent assortment. So what does this law of independent assortment state? Whenever two pairs of traits are combined, two pairs of traits are combined. Two pairs of traits means what? One pair of trait is one character. Two pairs of traits means we are considering two characters. Right now, that's what we did in dihybrid cross. Whenever two pairs of characters are combined in a dihybrid cross, the segregation of one pair of characters, the segregation of the alleles responsible for color does not depend on the segregation of characters that is responsible for shape. Color and shape are not dependent on each other. Color alleles assort independent of the shape alleles. If color and shape alleles were linked and they were dependent on each other, we would not have gotten new combinations at all. Now that we have obtained new combinations in the F2 generation, that indicates that the gene for color and gene for shape are not linked, they are not attached. Okay, so they independently get segregated during gamete formation. So whenever two pairs of traits are combined in a hybrid, segregation of one pair of characters, for example, segregation of the character responsible for color is independent of the other pair of character, which is the shape. If they were dependent, we would not have gotten newer combinations. If yellow color to get inherited, if it was always associated with round shape, then yellow could not have become wrinkled. Round would not have become green. The fact that we have obtained newer combinations that was not seen in the earlier generations indicate that when these pairs of factors are segregating during gamete formation, they are assorting independent of each other. This is the third law of inheritance. That's why it's called the law of independent assortment, where one pair of alleles don't really depend on the other pair of alleles during gamete formation. So that's about Mendel's dihybrid cross. Basically, we learn about Mendel's monohybrid and dihybrid cross in this chapter. So Mendel, after working for 7 long years, he performed so many experiments with pea plants. He had a sampling size of thousands, but he published his results in the year 1865. But back then, when his results were published, it did not receive the kind of recognition that it should have. There are some reasons why his work failed to be recognized when it was published. What are those reasons why his work remained unrecognized for a very long time? We'll try and understand that. There are 4 reasons. The first one is that communication was not very easy at that time. This was in the 1800s, right? You know how communication was not as communication is today. Today, somebody sneezes somewhere and then that can go viral for no reason, and the people across the globe would have watched that. So you have social media, you have so many media houses, and publishing anything is not a big task at all. But back in the day, in the 1800s, communication was not easy. His ideas to reach a lot of people, it was not easy. That's the first thing. Secondly, he described his concept of factors as discrete units. He said they are discrete units, they are distinct. However, it is there in the parent, exactly like that is going to get transferred to the offspring unchanged, unaltered. But many people who were into the field of genetics back then did not kind of buy this idea, because you see how there is so much continuous variation in nature. If such discrete factors were present, if they were unchanged, then how does one explain all of these variations in nature, right? So that was the second point. His concept of genes or factors as stable discrete units that control the expression of traits and the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature, right? So if that is the case, if a character, if an allele got passed on to the next generation as it is unchanged, then how is there variation? Like one dark person gets married to a fair skinned person, they have a child that is brown skinned, let's say. Okay, so how do you explain that? The parent is, the offspring is showing something intermediate. In nature, you see many such cases where there is no stark difference between two traits. It is continuous variation with respect to our skin color, our height, all of that. So if they were discrete factors, then how do you explain all of that variation was a question. That's why many people did not accept it. Thirdly, he used mathematics in biology, which was alien to a lot of biologists at that time. See, generally, even today, if you take people who study biology, don't really study math, and people who are good at math don't really understand biology very well. So, there's very little intersection between biology and mathematics as such. In physics, you learn about math, in bio, you learn about chemistry, but math and biology, unless you're talking about certain growth rate or certain graphs, you don't really incorporate math into biology that much. So even then, people did not accept the usage of biology in mathematics, they kind of couldn't make sense of it. Okay. So Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Basically, he was way ahead of his time. People failed to understand what he was capable of and what his studies meant. But it is unfortunate. He was a genius in statistics and mathematics and physics as well. So he could kind of think about using statistical analysis to understand the phenomena in genetics, but many people, many biologists back then couldn't quite make sense of it. That is the third reason. The fourth reason is that he said there are factors. Okay, we all have factors. Every character has a pair of factors. Okay, fine. I get it. I get your point. I will understand. I mean, I will try to take it. But show me where these factors are. People demanded for physical proof of those factors. Where are they present? What is their chemical nature? Is it present inside the cell? What is it made up of? Like, show me proof that these factors exist inside gametes, inside of our cells, then maybe I'll buy your theory. But he could not provide physical proof for factors. Where they are present, he did not know, because back then people had not worked out cell division. Microscopy was not so advanced. He did not know about genes or chromosomes or anything. So his idea was that there is something, but he could not show the proof that inside the cell, inside the nucleus, inside the chromosome, there is a gene, there is a nucleotide sequence. That is what factors are. He could not show the proof, physical proof for it, which is why people kind of did not accept his idea of factors. But nonetheless, better late than never, in the year 1900, three people independently discovered Mendel's work. They rediscovered Mendel's work. Okay, so back when it was published, not much, recognition. But many years later, three individuals, Hugo de Vries, Carl Correns, and Schermack, these three individuals, they were working in their own field of research, basically about genetics, and they happened to chance upon Mendel's work. And they were like, wow, this is great, right? You know about Hugo de Vries, right? You will learn about it in the chapter Evolution, about him in the chapter of Evolution. He is also credited for that saltation theory, right? He said that newer species can arise from a single large step mutation and all of that. It's the same genius, Hugo de Vries, wonderful, wonderful brain there. And Carl Correns and Eric von Schermack, these are the three people that rediscovered his work in 1900. So around the time 1900, Mendel's work got recognized again. It started to get recognized again. People started reading about it again. Around the same time, in the year 1902, specifically, microscopy was so advanced that people had kind of worked out how chromosomes behave during meiosis. During gamete formation, what exactly happens inside a cell? Before the cell divides, it's going to make an extra copy of DNA. When the cell divides during meiosis, during first meiotic division, homologous chromosomes separate. During the second meiotic division, sister chromatids separate. So all of these were worked out by scientists. So people kind of had an idea that within the cell, chromosomes are present and they behave like this. Okay. This was done by a different set of scientists, not these three guys. These three guys brought Mendel's work back into the picture by rediscovering it and talking about it and publicizing it. Around the same time, two other people, Sutton and Boveri. Okay? Walter Sutton and Theodore Boveri. These two scientists were working together. They got to know about Mendel's work from these three people's publicizing it. They openly spoke about it, and that's how they got to know about Mendel's work. By that time, they also knew that how meiosis happens had been worked out. They kind of correlated the two things and put it together. They thought about both of these things parallelly. Like, they used the movement of chromosomes, how that happens, to explain how Mendel's laws worked. Okay, think about it. What did Mendel say? Mendel say that in his first law, he said that factors occur in pairs. In a pair of dissimilar alleles, one will dominate, one will not, and that is called recessive, right? So that is Mendel's first law of dominance. Just like how Mendel said factors occur in pairs, in diploid organisms, they noticed how chromosomes also occur in pairs. This is from microscopic studies, they know. Second law of inheritance, what does it say? Chromosomes segregate during gamete formation, such that each gamete receives only one factor. Okay, factors segregate, Mendel has said. They noticed from chromosomal studies, from the study of meiosis, that even chromosomes are segregating during meiosis. Factors segregating, Mendel said. He did not know where these factors are. He failed to provide the physical proof, but these guys noticed that chromosomes are also segregating, similar to how Mendel said. Third law, what does it say? Factors segregate independent of each other. That is the law of independent assortment. What these guys observed was one pair of chromosome while segregating did not depend on another pair of chromosome. So when segregation of chromosomes were happening, that also happened, what do you say, independently. So they kind of put these two together and told people that, look guys, Mendel was talking about factors, poor guy, he did not know where the physical location of these factors was. He could not provide proof for the existence of factors. But today I'm going to tell you, the factors that Mendel was talking about back then is actually the genes located on our chromosomes. So they kind of provided physical proof for the existence of factors within the chromosomes, and they called it the chromosomal theory of inheritance. It's not like they discovered about how chromosomes move. They did not work on how meiosis happens. That was already done by a different set of researchers. They did not rediscover Mendel's work. That was done by 3 other scientists that I just mentioned. They just correlated the two things that were happening at around the same time in the early 1900s, and they put forth what is called the chromosomal theory of inheritance. So Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. So whatever chromosomes are present, if they are segregating, then along with the chromosomes, whatever factors or genes they are carrying, that is also obviously going to get segregated, right? So if you're sitting on a scooter and somebody is riding, if the scooter takes a left, you will go left with that because you're sitting on the scooter, right? So chromosomes are containing those factors. If the chromosomes are segregating, then the factors present in the chromosomes will also obviously segregate, right? Now, so basically Sutton united the knowledge of chromosomal segregation along with Mendelian principles, and then he called it the uh, chromosomal theory of inheritance. Now, whether this chromosomal theory of inheritance is true or not was verified by a person called Thomas Hunt Morgan. Now, this Thomas Hunt Morgan wanted to know whether the genes, that factors that Mendel was speaking about, is actually present on the chromosome. So, he performed some dihybrid cross experiments using fruit fly. What is a fruit fly now? Fruit fly is this fly that you see near ripened fruits. You can see that near curd. It looks like this. It looks like a house fly but smaller in size. So this is called the fruit fly. He performed many experiments using the fruit fly. The scientific name of the fruit fly is Drosophila melanogaster. Now this fruit fly was specifically chosen by Thomas Hunt Morgan for some reasons. What are the reasons why Morgan decided to, select this organism as his experimental organism? We'll see. Firstly, very similar to why Mendel selected the pea plant, the reasons are pretty much the same. It does not require too much work to take care of these fruit flies. They can be grown on a very simple synthetic medium like this in the lab. There will be many jars and bottle sorts of thing to culture these fruit flies. So a simple synthetic medium is used to grow them. They complete their life cycle in about 2 weeks. When you're learning genetics, I told you that it's important that the organism's lifespan is short so that we get offspring very soon. So they complete their life cycle from egg to larva to pupa to adult in 15 days, in about 2 weeks, which is great for genetics experiments. Similar to how one pea plant will produce many seeds after reproduction, here also after just one mating, many flies will be produced, many eggs will be produced. Next, in pea plant, the flowers were bisexual. Here, there is difference in sexes. They are unisexual. They are either male or female, but you can clearly differentiate between which is male, which is female very easily under a small magnifying glass also. So they are sexually dimorphic. By looking at its external features itself, you can say which is male, which is female. And they also, similar to pea plant, had a lot of characters that were clearly distinguishable. There were many hereditary characters that could be seen under low power microscopes and magnifying glasses, which did not involve fancy equipment. So since these were easy to work with, he chose Drosophila as his experimental organism. Now, I said that Morgan carried out dihybrid crosses. So when somebody is performing a dihybrid cross, obviously we're looking at two characters at a time, right? The two characters, he performed many dihybrid crosses using many characters. But one such example I'll tell you. For one dihybrid cross, he chose two characters, both of which were color based. One was the color of the body and one was the color of the eye. Okay. So in one of them, just a minute, a brown color, okay, somewhat brown. Is it visible? Yes, it is. Okay. So the body color could have been yellow or it could have been brown. The eye color was white or red. Over here, yellow body and white eyes, these are considered to be recessive traits. These were recessive. Okay, over here, the term recessive is not used. Instead, mutant, they use the term called mutant. Because of mutation, this recessive allele has formed. Brown color body and red color eyes, these are considered, these were dominant traits. But here we do not use the term dominant, instead we use the term wild type. That means this was present in the wild. Because of mutation, we got this particular type of allele. So when he chose organisms for his experiment, he made sure that one organism, one parent had a combination of yellow and white, and the other parent had a combination of brown and red. Okay. So if one parent, see here, one parent has yellow body with white eyes, the other parent has brown body with red eyes. This mutant or recessive phenotype, he considered a female that showed both recessive traits, and he chose a male that showed both dominant traits. See, the first step that you should do in a dihybrid cross is select parents that are true. That means they are homozygous. Okay. So he selected females that showed both recessive and he selected males that shows both dominant. This is the phenotype. Speaking of its genotype. Earlier, what did we do? If tallness is dominant, you take the dominant allele to be capital T and recessive allele to be lowercase t. But here, we do not use the letter of the dominant allele of the dominant trait. We use the letter of the recessive allele. So here, if this is yellow bodied white eyed, here the genotype of this will be YYWW. YY for yellow body, WW for white eyes. In case of males, brown body will not be B. Instead, it will be Y with a superscript plus. So this will be Y plus W plus W plus. So here, the genotype of this particular fly will be Y plus W plus W plus. What Morgan already knew was that the genes for both these characters were present on the X chromosome. In Drosophila, females have 2 X chromosomes, males have 1 X and 1 Y. So if this female has 2 X chromosomes, on each X chromosome there will be 1 copy. So this will be the genotype, right? Over here, the male has only 1 X chromosome. So there will only be 1 copy of each of these alleles. It will not be diploid. Instead, it will be diploid, but it will not have two alleles for each character. It will have only 1 allele for each character. Because only 1 X chromosome is there. These alleles are not present on Y chromosome. They are present only on X chromosome. Since males have only one X chromosome copy, you have only one copy of each allele. Females have two copies of X chromosome. So we have two alleles on each, for each character you have two alleles. If only it was present on any autosome, then both males and females would have had two copies of each allele. Okay, so this is like the background for the experiment. Okay, how the alleles are represented, which is dominant, which is recessive, which is wild type, what is a mutant, all of that I've told you now. Now what they did in this experiment was very similar to what Mendel did. They took female that was showing both recessive traits, male that was showing both dominant traits, they crossed them. After that, what you obtain is the F1 generation. Previously, what did Mendel do? He self-pollinated the F1 generation. But here, can you self-pollinate it? You can't, because they are not unisexual. They are they are not bisexual, they are unisexual. So what they do here is called intercrossing. In intercrossing, whatever offspring you obtain in the F1 generation, one male and one female from that, they are crossed. Okay, so you can't self-pollinate here because they are unisexual. So intercrossing is done, and as a result, what we obtain is called the F2 generation. Okay, now when they perform this cross, this is the results that they obtained. So you can see over here, this is the cross that I'm talking about. You can see these rod sort of things. What they are, how I will help you understand. To the left side, you have the female parent. To the right side, you have the male. The female has two X chromosomes and the male has one X and one Y. This hook sort of thing is the Y chromosome. It's not relevant to us over here because we are speaking of genes that are present specifically on the X chromosome. I mentioned how the genotype here will be YYWW, the same thing they've represented on top of a chromosome. Instead of writing like this, they've shown it on the chromosome. Earlier, we did not know that the location of factors was chromosomes, so you would write it as in letters. But now, after the chromosomal theory of inheritance, we are told that these genes or factors are located on the chromosome. We've represented it on the chromosome. So here at this location, you have information for body color. At this location, you have information for eye color. Here also on the X chromosome at the same location, you have information for body color. Over here, you have information for eye color. Okay. Now, what happens after you cross them is, in the F1 generation, this is the F1 generation, you will obtain females that are wild type and males that are mutant, like mother. Here, mother is showing recessive, father is showing dominant. But in the first generation, daughters will be like father with respect to phenotype, sons will be like mother with respect to phenotype. Why? Because sons will receive their X chromosome from the mother. Mother had recessive allele, same thing is passed on to the son, so the son is showing mutant phenotype. Females will receive one X from the mother, one X from the father. Since they have the dominant alleles in the X chromosome of father, they got one copy of the dominant allele, they will display the dominant phenotype. All of the females will be heterozygous like this, displaying wild type phenotype. All of the males will display the phenotype, recessive phenotype of the mother. Now, when you intercross them, you obtain what is called the F2 generation. In the F2 generation, you get few parental types. Okay, parental type means same combination. Yellow body with white eyes or brown body with red eyes. That is the parental combination. And you get a few recombinants as well. What are recombinants? Recombinants are, recombinants are yellow body with red eyes or brown body with white eyes. Okay, so here you see these are wild type. Here in parental type you have yellow bodies with white eyes, parental combination, or you have brown bodies with red eyes. Recombinants will be yellow body with red eyes and brown body with white eyes. Of course, it does not look like a Drosophila, I know, this is just for representation. So these are the recombinants. These combinations did not exist earlier. Earlier, yellow was always associated with white, brown was always associated with red eyes. But now we got newer combinations. But if you look at the percentage of how many were parental types and how many were recombinants, he noticed the huge deviation from Mendel's dihybrid cross ratio. In Mendel's dihybrid cross ratio, you got 10 out of 16 that were, parental type. 6 out of 16 were recombinants, right? This was the ratio. But if you see here, around 98.7%, almost 99% were parental types. Very few, just about 1.3% were recombinants. So this was a deviation from Mendel's dihybrid cross ratio. In Mendel's dihybrid cross ratio, we got parental type 62.5%, that is 10 out of 16. Recombinants we got 6 out of 16, that is 37.5%. But what did we get in this cross? We got 98.7% parental type, 1.3% recombinant, which is a huge deviation from Mendel's 9:3:3:1 ratio. Now, that is one set of experiment that Morgan performed. They performed another experiment, similar experiment using fruit flies only, using two other different characters. One character here again was eye color, and the other character was wing size. Okay. So when they chose these two characters in the dihybrid cross, they noticed that the parental types were 62.8% and recombinants were 37.2%. Eye color, like we already know, could be white or red. Wing size could be small, in which they call miniature, or it can be normal. The normal one is wild type, the miniature one is mutant. Okay. So they performed a similar cross. In this cross B, they noticed how the ratio was similar to Mendel's dihybrid cross ratio. 62.8% is close to 62.5%, 37.2% is close to 37.5%. So depending upon which characters they chose, how much percentage of parental types and how much percentage of recombinants they obtained, it varied. Okay. So based on their results, what they said was, whenever genes are located on the same chromosome, they are physically linked. Okay. So if this is a chromosome is made up of DNA, right? So if you unwind the chromosome, you have DNA like this. There could be one gene present here, one gene present here. In this segment, there is one gene. In this segment, there is another gene, we can say. But if you see, they are physically attached to each other. They are part of the same DNA molecule. So they are physically linked to each other. The fact that the genes present on the same chromosome are physically linked to each other, he coined a term to call it linkage. This describes the physical association. It's like they're physically attached to each other, right? So that is, he used the term linkage to describe that physical association. Now what he says is, notice the difference between crosses A and B. In cross A, the two genes that he considered were on the same chromosome. Even in cross B, the two genes that he considered, the two characters that he considered, had genes located on the same X chromosome. But look at the position of genes here and here. Here the genes are located very close to each other. Here the genes are located distantly from each other. So what he says is, whenever the genes are close to each other, their linkage is very tight. Whenever genes are located distantly from each other, they are still linked only, but their linkage is quite loose. Okay. For as long as gene is located on the same chromosome, they are physically linked, but how tightly or loosely they are linked depends upon the distance between them. Whenever genes are located close to each other and they are tightly linked, the chances of getting recombinants is very less. Whenever the genes are located on the same chromosome but distantly located, not too close to each other, then the chances of getting recombinants is much higher in comparison. Here, because they were so close to each other, you got only 1.3%. Here, since they were distantly located, you got as much as 37.2%. Now, he coins another term called recombination. which kind of describes how non-parental gene combinations are produced. Non-parental means recombinant, newer combinations. Okay. So, like I mentioned, Morgan and his co-workers, they already knew that all of the genes they were considering were present on the X chromosome. When the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than non-parental type, which is low. So he explains that using the concept of linkage and recombination. Even when genes are grouped on the same chromosome, some genes are tightly linked, like I said, very close to each other, and few other genes are loosely linked. For example, in cross A, they were tightly linked, in cross B, they were loosely linked. If they are tightly linked, the recombinants percentage will be less. If they are loosely linked, the recombination percent is more. Now, why is that so? How can you say whether it is tightly linked? How can you say that if it is tightly linked, they are there are less chances of recombination? See, we have to go back to our uh, meiosis to understand this. What exactly happens during meiosis? How are new variations formed? It's formed because of a process called crossing over that takes place during a packet in substage of meiosis, right? So what happens then is homologous chromosomes. Let's say this is one X chromosome and this is another X chromosome. Okay, since they are homologous chromosomes, pairing up and crossing over will happen between the two of them. So let's say on this chromosome you have information for what is that, eye color and mm, body color, right? Yellow bodied, white eyes. Here you have a brown body, red eyes. Okay, so let's say this is the case. When crossing over happens, it's not that specific genes will get crossed over. Segments of chromosomes will get crossed over. So when crossing over happens between these two, it's possible that this entire segment has gotten crossed over with this segment. So within the same segment, both the genes are present, so they're getting crossed over together. Even though crossing over has happened, W plus has not come to W has not gone with W plus because it is getting crossed over as a segment. If only the genes were located distantly. Let's say there was one gene here that is responsible for wing shape. Okay, so this one had miniature wings, this one had normal wings. So if in this segment crossing over happened, okay, so we are looking at two characters here. One character is let's say the color of the eyes and wing shape. If we are looking at this, if crossing over happened here, then since these two are located distantly from each other, it's not likely that they're going to get crossed over as a single unit, right? So the chances of newer combinations forming are much higher when it comes to these two genes. So if crossing over happens in this place, then we are going to get a new combination of alleles, a new combination and a new genotype. So if crossing over happens here also, we are going to get a new genotype. So when they are distantly located from each other, the chances of getting newer combinations is much high because they are not going to get crossed over as a single unit. Okay, so that's how it is. Now there's this one person called Alfred Sturtevant. Sturtevant, that's his name. A little difficult to pronounce. So he also was working along with Morgan in his lab and he kind of figured a way to create genetic maps based on the recombination frequency. So let's say in this particular cross, you got 1.3% recombination. That is the recombinant frequency here. So he said that on this chromosome, there is 1.3 units of distance present between these two. Based on how many percentage of recombinants he got, he kind of started plotting the position of genes on chromosomes, and that was the first kind of genetic maps to ever be created. Okay, so Morgan student Alfred Sturtevant, He used the frequency of recombination between gene pairs present on the same chromosome as a measure of the distance between them. Obviously, if the recombination frequency is less, that means they're close to each other. If the recombination frequency is more, that means they're distant from each other. So based on that, he mapped it, okay, or mapped the position of genes on the chromosomes. So that's about linkage and recombination. Now let's learn about what is called polygenic inheritance. Poly means what? Poly means many. Many means how many? More than 3 or 3 or more than 2 basically. So if any one character is controlled by more than 2 genes, a minimum of 3 genes, then we call it polygenic inheritance. Okay. So yeah, such genes are controlled by 3 or more genes, minimum 3 or more than 3 genes. Then we call it polygenic inheritance. In Mendel's experiments, we saw how each character was controlled by one gene. But it's possible that certain characters can be controlled by many genes, as many as 3 genes. In that case, we call it polygenic inheritance. There are two important examples for polygenic inheritance, one being the skin color in humans and another being the height in humans. If you look at this picture, it has a group of humans. You can see how there's a gradient with respect to height. Okay, so there are a little bit short individuals, slightly more taller than her, slightly more taller than her. Unlike how we saw in the height of pea plants, it was either 6 feet or higher or 2 feet or lesser than that. That gradient, that stark contrast, you don't see in humans. There are people who are 5 feet tall, there are people who are 5 feet 3 inches tall, there are people who are 5 feet 8 inches tall, there are people who are 5 feet 5 feet 10 inches tall, so 6 feet tall. So there's a gradient, right? Same goes with skin color. You can't say violet white like we did in plants or green or yellow like we did in pea plant. Even in the color complexion of humans, you see a huge gradient. So mostly these characters are controlled by a minimum of 3 genes. That's why we call it polygenic inheritance. In such inheritance patterns, you will notice how the trait has environmental influence as well. For example, let's say both my parents are super tall and I have it in my genes to be tall too. But if I don't consume the right amount of nutritious food, if I'm not eating sufficiently, if I'm not consuming the nutritious food, then just by having genes, I will not be able to grow tall. For growth to happen, nutrition is equally essential, right? So that way it can have like an environmental influence, and the kind of food that you eat can influence your height. Speaking of skin color also, just because you have genes that are, that make you fairer complexioned, does not mean you will be fair, because it has the influence of how much time you spend outdoors. If you're too much in the sun, then melanin production will be triggered in your skin and you will become, more melanin is produced, you will turn darker. So these traits, usually polygenic traits, they have environmental influence. Okay. Now, in this chapter, we're going to learn about human skin color as an example for polygenic inheritance. The human skin color is believed to be controlled by 3 genes called A, B and C. All of those genes can either exist in their dominant forms, which is capital A, or recessive forms, which is lowercase a. Capital A, capital B, capital C are the dominant forms, and the lowercase a, lowercase b, lowercase c, these are the recessive forms. If it is in dominant form, regardless of which gene, whether it is A, B or C, if it is in dominant form, that will trigger melanin production. If it has recessive alleles, lowercase alleles, then melanin production is not triggered. So basically, more number of dominant alleles, more darker will be the complexion, less number of dominant recessive alleles, or more recessive, less dominant alleles, more lighter will be the complexion of individuals. Okay, so let's assume that the 3 genes that control skin color are A, B and C. Its dominant forms are capital letters, lowercase s, recessive. Dominant triggers melanin production. Melanin is a pigment that will give you color to the skin. Okay, so darker people have more melanin in their skin. Lighter complexion people will have fewer melanin pigments present in the skin. Okay, so people who have all dominant alleles will be the darkest, will be of the darkest complexion. People who have all recessive alleles will be super pale. Okay, so in between you can see intermediates. If they have 5 dominant and 1 recessive, they'll be slightly lighter than this. If they have 4 dominant and 2 recessive, they'll be slightly lighter. So depending upon how many dominant alleles are there, the color, the complexion of the skin will vary. More number of dominant alleles, more darker will be the complexion. Regardless of what is, whether you are dark or light, I mean, in this generation, you should not worry too much about that, especially in India. There's a lot of bullying that happens for people who are dark complexioned. Of course, Indians are brown, but still we tend to look down upon people who are dark. So, I mean, just darkness does not mean inferior, and being fair does not mean superior. At least you all should, consider that and not bully people for their skin tone. It's not something that they chose. It's not something that is bad or evil. It's just people who are fair are just, they got lucky with their combination of genes. It's not like they worked hard for it or anything. It's just considered conventionally beautiful. Otherwise, it's not like it has any additional benefit or anything. So yeah, all colors are okay. All colors are beautiful, right? Now, another thing that I want to point out, when I say 5 dominant alleles, I don't specify which are the alleles that are dominant. It could be capital A, capital A, capital B, capital B, capital C, lowercase c, or it could be capital A, lowercase a, capital B, capital B, capital C, capital C. Any 5 of the 6 alleles could be dominant. Here, which one is dominant, A, B, or C, does not matter. Quantity of how many dominant alleles are there, that is what matters. So that's why this is also called quantitative inheritance. More number of dominant alleles, more the effect will add up. Okay. Yeah. So that's about polygenic inheritance. Now let's speak of pleiotropy. Pleiotropy is kind of like the opposite of polygenic inheritance. So what happens in pleiotropy is one gene is responsible for many phenotypic effects. We saw in Mendel's studies, one character, one gene. One character is controlled by one gene. In polygenic inheritance, we saw one character is controlled by a minimum of 3 genes. In polygenic inheritance, we are saying that one gene controls many phenotypic traits. The gene number is 1, but the number of traits it's controlling is many. That is called pleiotropy. One example for pleiotropy is a metabolic disorder called phenylketonuria. In short, it is called PKU. So people who suffer from this disorder, phenylketonuria, they have a defective gene, and that gene was responsible for the production of an enzyme called phenylalanine hydroxylase. In short, it is called PAH. Okay. So this gene is mutated. Since this one gene is not functioning, it shows many phenotypic effects. More about phenylketonuria we'll study later also. Just because of that one gene, you can see the effect on skin color, you can see the effect on hair color, you can see the effect on brain development also. Individuals who have just one mutated gene, who have that mutated gene, that copy, who suffer from the disease because of that one gene, what happens is their skin color is reduced. They are very pale in color, reduced skin pigmentation. Their hair pigment is also reduced and the hair volume is also less. They have very thin hair and also they are mentally retarded. Okay, so these are the different phenotypic effects that one gene has. That is an example of pleiotropy. That's all about the various patterns of inheritance. Now let's get started with sex determination. Let's begin with sex determination in humans. Sex determination is basically how exactly the gender of a child, of an offspring, is decided. There are various ways in which sex determination can happen. Right now in humans, we are going to learn about how chromosomes determine the sex or the gender of an offspring. Okay. So in humans, we have a total of 46 chromosomes. Human males have 44 autosomes plus one X and one Y chromosome, while the females have 44 autosomes plus 2 X chromosomes. So the males during spermatogenesis undergo meiosis and produce sperms that are haploid. So the sperms can be of two types. 50% of the sperms will contain 22 autosomes with an X chromosome. The remaining 50% will contain 22 autosomes with a Y chromosome. Whereas the females, when they undergo oogenesis, meiosis takes place But since they have the same kind of sex chromosome, both X, all of the X will have 22 autosomes plus an X chromosome. Now here males produce two types of gametes, therefore males are said to be heterogametic. The reason is because they have two different types of sex chromosomes. Females produce just one type of gamete, which is why they are said to be homogametic. Over here, the gender is decided by the sperm. If the sperm carrying the X chromosome fuses with the egg carrying the X chromosome, then it will result in a zygote that has 44 autosomes plus 2 X chromosomes. This is the female baby, a baby girl. If the sperm carrying Y chromosome ends up fertilizing the egg carrying X chromosome, the baby will end up having 44 autosomes with an X and a Y chromosome. This will be a male offspring. So, whether the baby is male or female does not depend on the egg. It does not depend on the female gamete because all female gametes are alike. But the male gametes either carry X chromosome or Y chromosome depending upon the sperm that is carrying the X chromosome or Y chromosome, the gender of the baby will be determined. If it carries the X chromosome, it's going to be a girl. If the sperm is carrying the Y chromosome, then it's going to be a boy. So it's entirely dependent on the father, the gender of the child. Now, speaking of sex determination in some other organisms, it's not necessary that every organism there should be, in every animal, sex is determined by X and Y chromosomes only. Let's see how it happens in other organisms. In Drosophila, similar to us, females are homogametic. Females have two X chromosomes while males have one X and one Y. So again here males are heterogametic. Another example of male heterogamety can be seen in grasshoppers. Here the case is a little different. It's not like the males have two sex chromosomes, two different types like X and Y. There is only one X chromosome. The sex chromosome is only one type, X chromosome. Females have two copies of that, whereas males have only one copy of that. This is called exo type of sex determination. O is not another chromosome like Y. O means absence of the sex chromosome. It indicates that male grasshoppers have only one sex chromosome, X. Okay. So what happens here is males will have one chromosome less than females. The female gametes will carry, the egg will carry X chromosome along with the autosomes. Okay, so I'll write autosomes plus X. Whereas the sperm will carry X chromosome with autosomes, or the sperm will only have autosome. If the sperm that has only autosomes fuses with the egg, then that will be a male. If a sperm having X chromosome fuses with the egg, then it will be a female. Okay, so here again, males are producing two types of sperms, two types of gametes. That's why it's said to be male heterogamety. Is it the case in all animals? Are males always determining the gender? Are males always heterogametic? Not really. If you see a fowl like chicken, you can see the females will produce two different types of gametes. Over here, the males have sex chromosomes called ZZ. Two copies of the same sex chromosome you can find in males. But females will have two different types of sex chromosomes, Z and W. So over here, the egg will either have Z along with the autosomes or it will have W along with the autosomes. But all of the sperms will have Z only. So here the sex is determined by the female. If the sperm fuses with the egg containing Z chromosome, it will become male. If it fuses with the egg containing W chromosome, it will become female. W chromosome is not found in males, it's found only in females. Okay. So that's about how sex determination happens in some other animals. Now it's interesting how sex determination happens in honeybees. This is called haplodiploid type of sex determination. Over here, the number of sets of chromosomes in an organism determines its sex. Here, in a honeybee hive, there is a queen bee that is the only fertile female present. The queen bee is diploid, has 32 chromosomes. There is no separate autosomes or sex chromosomes. The total number of chromosomes is 32. And the males, the drones, are haploid. They have only 16 chromosomes. So drones are males and queen is female. Each hive will have only one queen, and the other queens, the other females will be workers, but they are not capable of reproducing, they are sterile. So this queen will undergo meiosis to produce eggs that are haploid. While the sperms will undergo mitosis, the males will undergo mitosis to produce sperms that are haploid. Now, once in a while, the queen goes on what is called a mating flight. So, once she goes on a mating flight, there is a sac sort of structure in the queen's body where she collects the sperms of the males. Depending upon the need of the hive, whether it requires more workers or more drones, the queen will decide whether the hive needs more females or males. Okay. So, if the hive needs more males, more drones, then she will fertilize. She will use the sperms that she has collected. It will be viable for a very long period of time. Those sperms she will use to fertilize her eggs, and that will result in the formation of females. Okay. So, this will be 32 chromosomes. This will become female bees. If she needs more drones, then the egg will directly develop into drones that are male. The process by which egg alone will develop into an offspring without any interference from the male parent, this is called parthenogenesis. So if you think about it, these male honeybees don't have a father at all because they are formed from the female. But the female honeybees will have father and mother because for the female to form, you need contribution from the sperm as well as from the egg. So males, it is said, they do not have fathers, but they can have grandfathers. How is that? The females will have a mother. The queen will have a mother. Queen will have a father, right? Females will have father. So the mother's father is the grandfather for male drones. They themselves do not have fathers, but they have grandfathers through their mother. They can never bear sons. They can never have male children because for formation of male children, sperms are not involved at all. Egg will directly give rise to the male children. But they can have grandsons. They cannot have sons, but they can have grandsons. So if they've contributed to a female and that female becomes a queen and that queen gives rise to a drone, then they will have, they may have grandsons, but they can never have sons. So a honeybee drone does not have a father, but can have grandfather through the mother. They can never have sons, but they may have grandsons through their daughters. Okay. If the female bees become queens, then there is a possibility. So that's about sex determination in honeybees. Now let's speak of mutation. Mutation is a change in the sequence of nucleotides in DNA. It can happen because of many reasons. So it is a phenomenon which results in the alteration of DNA sequence. Mutation is one of the reasons why there is variation in a population. Of course, sexual reproduction brings about newer genetic combinations, but mutation also gives rise to newer combinations. I told you, right, that's how alleles form. Newer alleles arise in a population because of mutations itself. Now, mutation can happen at the nucleotide level in DNA where a nucleotide base pair gets changed, or it can happen at the chromosome level itself where the chromosome, one segment of the chromosome gets broken. It gets broken, becomes inverted and attaches. It gets broken and goes and attaches to some other chromosome. So, at the chromosome level also, mutations can happen. Okay. So, whichever factors induce mutations, cause mutations are called mutagens. Muta refers to mutation, gen refers to generating. Any agent that is capable of generating, causing mutations in our body are referred to as mutagens. These mutagens can be physical, chemical, or biological agents. Example for physical agents include UV rays, X-rays, and all of that. They are X-rays, especially they are ionizing radiations, and they can cause damage in DNA. They can cause mutation in DNA. Speaking of chemical agents, you may have seen on cigarette packets and even watched these ads where it is told that cigarette smoking can cause lung cancer and oral cancer and all of that. So how does cancer get caused? It's because of mutations in certain genes itself. So there are many chemicals present in the cigarette smoke that are capable of causing mutations in our body. So chemicals in cigarette smoke are considered as chemical mutagens. And speaking of biological agents, there are certain viruses which are capable of bringing about changes in our DNA. They are the biological agents or biological mutagens. Okay. Now there's something called pedigree analysis that we must learn about. So whenever you study any trait in humans, let's say, it's not easy for you to perform crosses and experiment like you do with animals and other plants, right? So in order to understand how any trait is getting passed on across generations, what inheritance pattern is it following? Is the gene present on an autosome? Is it present on a sex chromosome? How exactly is it getting expressed? If you want to study all of that, we can do that by what is called pedigree analysis. Pedigree analysis is generally done in order to study genetic diseases, Mendelian disorders, but it can be done for any trait. Like it need not necessarily be any disease. It can be done for any trait. Like how is the hair color getting transmitted? Or some people have polydactyly. They have more than 5 fingers, like Hrithik Roshan. How does that get transmitted across generations? All of that you can study using pedigree charts. So in these pedigree analysis, there are a couple of symbols that we should know. We use symbols to represent whether uh, we use symbols to represent the family tree. Basically, we are studying about genetic disorders or traits. So we are learning about how it got passed on from one generation to another. So all of those generations we will represent using certain symbols. So here are the symbols that you should be aware of in pedigree analysis. The first one is male. Male is represented by a square, female by a circle. If you do not know the sex, if you're not sure of the sex, then it is a rhombus, a diamond shape. Now, if the male or female do not suffer from that particular disease, or if they are not showing signs of that trait, then you just draw the outline. If they are showing that phenotype, that particular phenotype is being displayed, or that particular disease symptoms are being displayed, then you shade that entirely, you make it opaque. Okay, so if it's shaded, that means those individuals are affected. If it's unshaded, if it's just the outline, that means it is unaffected. Okay, now whenever you want to show marriage or mating between two individuals, obviously it will happen between a male and a female. So one square and one circle, in between that you should draw a line. It need not be necessary that the male only has to be first, then female, nothing like that. If there is a line drawn between a circle and a square, that indicates mating. If mating has happened between two closely related individuals, then you represent that by drawing two lines between the two. Now, how does that matter? Why would it matter whether they are closely related or not? Basically, when closely related individuals get married, it's called consanguineous marriages. Okay. So you may have noticed how sometimes when individuals that are closely related get married to each other, the chances of them having children with developmental delays or any genetic disorders is very high. So basically, while we're studying inheritance patterns of any disorder or trait, that plays an important role. That's why that is also represented. Now, a couple, they've gotten married, they have some children. The children are represented like this in the form of a flowchart as the next generation. Over here, the number of children are represented according to their order of birth. If the first baby was a girl, then you first should be a circle, then it should be a square if the second one was a boy. If both of them are girls, then circle, circle. If first is a boy, then it's a girl, then first square, then circle. So based on the order of birth, you will represent that. If that baby is affected, that child is affected, you will obviously shade it. So over here, you're looking at the child being affected while both the parents are being normal. Okay, so more about how that happens and all we'll learn later. Sometimes if you think back on your ancestors while you're explaining to a geneticist what happened, you might not remember how whether the individuals in your family, like in the past generations, they were males or females. You just know, okay, that person had 5 children. I'm not sure about the sex, but none of them were affected. then you can represent it like this, like a rhombus with 5 inside, none of them are affected. Okay. Now, genetic disorders are of two types. If there is any mutation in a single gene, then that is called Mendelian disorders. One gene will be affected in Mendelian disorders. Because of one gene defect, it will result in a disorder. Now, uh, now the gene that is responsible for the disorder could be present on an autosome or it could be present on a sex chromosome. If it is present on an autosome, it is called autosomal disorder. If it's present on a sex chromosome, it's called sex-linked disorder. Now, the gene could, express itself. The disorder could express itself either in its dominant form or in its recessive form. If the disorder is, you see the disorder symptoms in its dominant form, then you call it autosomal dominant disorder. If it is expressed in its recessive form, then it's called autosomal recessive disorder. Likewise for sex-linked as well. If it's expressing in the dominant form, it's sex-linked dominant. If it's expressing in recessive form, then it's sex-linked recessive. We have examples for all of these. We'll learn about that. Not all of these. We have examples for recessive traits. We don't have examples for dominant. Autosomal dominant, I'll give you an example. We will not discuss that in detail. But myotonic dystrophy is an example for autosomal dominant trait. Like that, you have chromosomal disorders. Now, the chromosomal disorders are when there is change in the chromosome structure, chromosome number. Like normally we're supposed to have 47 chromosomes. If somebody ends up having, normally we're supposed to have 46 chromosomes. If a person ends up having 47 or 45, more or less number of chromosomes than normal, then such disorders are referred to as chromosomal disorders. In this chapter, we have to learn about both Mendelian as well as chromosomal disorders. We'll begin with Mendelian disorders. I'll show you how a pedigree works for Mendelian disorders. So like I mentioned, myotonic dystrophy is an autosomal dominant trait. That means the symptoms of myotonic dystrophy, for the symptoms to appear, there should be at least one copy of dominant allele. In this genotype or gene combinations or allelic combinations, myotonic dystrophy, you can see. If I'll write affected here, individuals that are affected will have at least one copy of dominant allele because it's an autosomal dominant trait. If it has both recessive alleles, then they are normal individual. Over here you can see whichever is not shaded, all of them, it's easy to guess, it is normal. But if they are showing signs of the disease, that means there are two possibilities. Either they can have capital A or they can capital A or they can have capital A lowercase. So here, if you look at this couple, this woman has myotonic dystrophy. Some of her children have myotonic dystrophy, some of them don't. Like this person has it, this son, this daughter has it. This daughter doesn't have it, this son doesn't have it. Yeah, they don't have it. They have 4 children. 1, 2, 3, 4. This son is married to this woman, this daughter is married to this man. You can see the line connecting the two. I hope it's visible. Okay. So, now, can you guess the genotype of the second child in the second generation or the first son in the second generation? If they ask you, see, this is very easy to predict. If it's not showing signs of a disease, that means it is recessive, homozygous recessive. Now, this can be homozygous dominant or recessive. Can it be homozygous dominant? If it was homozygous dominant, then every one of the offspring should have received a capital A from the mother. All of them should have shown the signs of the disorder. But here, you can see that all of the children are not affected. Some of them are normal, some of them are affected. If this individual, this son is normal, this daughter is normal, they should have lowercase a, lowercase a. They got one lowercase a from the father, but another lowercase a they should have gotten from the mother itself. No. So mother has a copy of recessive allele. Along with that, she has a copy of dominant allele. That is why she is showing symptoms of the disease. Same goes for these two. Both of them would have received recessive allele only from the father. Mother, unfortunately, has passed on the defective allele to them, so they are heterozygous. Here again, this person will be heterozygous. So here, you have to notice if it is a dominant trait, unless the parents are affected, offspring cannot be affected. Over here, you see one parent is affected, one child is affected. Here, one parent is affected, two children are affected. But look at this family. This son is not affected, wife is not affected, children are not affected. Children cannot be affected unless the parents are affected. Okay. So that is how it is in dominant disorders. In recessive disorders, that is not the case. Even if parents don't appear to show the symptoms of the disease, the offspring can be affected. Okay, so here is an example for an autosomal recessive trait such as sickle cell anemia. Okay, so here lowercase a, in the recessive state, they will show symptoms of the disease. If they are capital A or capital A lowercase a, they will be normal. If they are homozygous or heterozygous dominant, they will be normal. Only when they are recessive, they will show signs of the disorder. So whichever ones are affected, they all will be homozygous recessive. If they got two copies of the recessive allele, then both parents should have one copy of the recessive allele. That is how they got it, right? So both the parents are heterozygous. They have a defective allele, but they're not showing symptoms of the disease because they have one normal allele also. So if this is how it is, even when the parents are not affected, offspring are affected. Parents are not affected, offspring are affected. That can happen only in recessive disorders. Okay? Because in heterozygous condition, these individuals will appear normal. They will not show signs of the disorder. Okay? Now, let's speak of some Mendelian disorders, starting with color blindness. Color blindness is an X-linked recessive disorder. It is sex-linked. The gene responsible for color blindness is present on the X chromosome. Color blindness does not mean individuals will view the world in black and white. Color blindness has many types, red, green type, red, blue type, many types are there. Okay. Why does that happen? It happens because of a defect in the cone in our eyes. You know, in your eyes, you have many layers, and the innermost layer, there is retina. In the retina, you have photoreceptor cells, rods and cones are present. So, cones are responsible for the colored vision. In individuals that have color blindness associated with the X chromosome, which is called red-green color blindness, they fail to distinguish between red and green colors. What we see normally like this, it appears like this to them. I want you to notice how green does not appear green. It appears somewhat olive green or brown. Red does not appear red. Here is the red color. Red is also appearing different. And even orange here does not appear orange because orange is a mixture of red and yellow. Their cones that perceive red color is faulty. It is defective. It cannot perceive red color. In the absence of red, orange will only appear like yellow, right? So many colors in the color wheel, they will not be able to perceive normally. That is called red-green color blindness. Now this red-green color blindness is located, the gene responsible for that is located on the X chromosome. Let's say X allele is normal, XC is the defective allele. X with a superscript C is the defective allele. Now, in females, there are three possibilities of genotypes. Either there can be two normal alleles, in which case obviously their vision will be normal. They can have one normal allele and one defective allele. Even in this case, the vision will be normal, but they are called carriers because they themselves do not have color blindness, but they have a defective allele which they can pass on to the next generation. They are carrying a defective allele which they can pass on to their next generation, to their sons and daughters. Okay. Now, another possibility is both of them are defective. So, such individuals will be colorblind. For men, in males, since they have only one X chromosome, there are only two possible genotypes. Either they can be XY, which is normal, or they can be XCY, which causes color blindness. In males, especially in X-linked recessive disorders, there are no carriers. Carriers cannot exist. Carriers can exist only when they have two copies of that allele. One is normal, one is defective. But here, there is only one copy of the allele. So males here, since they have only one copy of the allele, they are said to be hemizygous. We don't call them heterozygous because they have only one copy of that allele. Whatever is there, that will express. If there is normal allele, they will be normal. They will have normal vision. If there is a defective allele, they will suffer from color blindness. That is not the case here. Here we have heterozygous condition. Heterozygous means two copies of the same gene for the same gene, but they are dissimilar. Then you can call it heterozygous. But here you can't call it heterozygous because they don't have two copies at all. If they have two copies and one was different from the other, then you can call it heterozygous. All they have is one copy. So we call it hemizygous. Okay, so whatever is there will express. Now, in order for a female to become colorblind, a colorblind female, this is the phenotype that should exist. Females get one X chromosome from the mother. Female gets the other X chromosome from the father. If father has to pass on the defective X chromosome to the female, then the father should have a defective copy. If father has a defective copy, the father is colorblind. If the mother has to pass on the defective X chromosome with the defective allele for colorblindness, she should have at least one copy of the defective allele. So, mother should at least be a carrier or she is also colorblind. So, for a female to become colorblind, father 100% has to be colorblind and mother should at least be a carrier. But that's not the case for males. If a male has to be colorblind, it does not matter whether the father is colorblind or not, because father does not even pass on the X chromosome to the sons. Father passes on the Y chromosome to the sons. The sons can get colorblindness only from the mother. So, even if the mother is a carrier, there is 50% chance for the child, for the son to become colorblind. If the mother is colorblind, 100% the son will be colorblind. Okay. So, if mother is carrier, there is 50% chance. If the mother is colorblind, then 100% the son is going to be colorblind. Because for males to become colorblind, even if the mother is a carrier, 50% chances are there. The occurrence of colorblindness in the human population is more seen in males than in females. For a female to become colorblind, father has to be colorblind, mother has to be a carrier. It's not common in nature that two colorblind people bump into each other and they'll be like, come on, let's get married. Right? That doesn't happen. So even if the mother herself is not showing symptoms of colorblindness, the son can get colorblindness. 50% chances there. That's why the occurrence is more in males. Okay. So colorblindness is a sex-linked recessive disorder. This occurs due to a defect in the red or green cone of the eye. They will not be able to distinguish between red and green colors. This happens because of the mutation of genes present on the X chromosome. This is more common in males, as much as 8%, but lesser in males, just about 0.4%. Why? Because the genes are located on the X chromosome. Females have two copies and males have only one. So the son of a woman who carries the gene, just she is a carrier, there is 50% chance. But if a daughter has to be colorblind, that means the father has to be colorblind and the mother at least has to be a carrier. So that's less likely in nature. That's why in females you don't find that much colorblindness. Now let's move on and talk about hemophilia. Hemophilia is a blood clotting disorder. This is also X-linked, sex-linked recessive disorder. The gene responsible for hemophilia is located on the X chromosome. Females have two X chromosomes, males have one X chromosome. So females can be normal carriers or affected, but males only two possibilities, either they have hemophilia or they are normal. Okay. Same here also, males are more affected. Females it's very rare, because when two defective alleles come together in the fetal stages itself, the I'm sorry, in the early embryonic stages itself, the pregnancy will be aborted. It won't be viable until later stages. So what happens here is, uh, for blood clotting, there are specific clotting factors present in the plasma, right? Soluble protein clotting factors will be present. You would have learnt about that in body fluids and circulation. When there is a wound, when the blood vessel breaks, how exactly does a clot form? There are so many proteins involved, prothrombin to thrombin, and fibrinogen to fibrin, and then a mesh of fibrin forms and platelets go and seal. Clotting factors, calcium ions are involved. All of that you study about, right? So basically one protein clotting factor that is essential in the process of blood clotting that will not be produced because of a defective gene present on the X chromosome. Present on the X chromosome. Just because of that one clotting factor, in these individuals, blood clotting itself will not happen, and very small cuts also can result in uncontrollable bleeding and people could even die. So that's why it's called hemophilia. And hemophilia is also called the royal disease because apparently Queen Victoria of England, she was a carrier of this disease, and many of the descendants in her family had hemophilia. Many of the men in her family had hemophilia. That's why it's also called the royal disease. Okay, so even over here, since it's an X-linked recessive disorder similar to color blindness, here also you will see that if a mother is carrier not showing signs of the disorder, 50% chance of the son becoming hemophilic, but it's very rare in females. As I mentioned, when two such defective alleles come together in the zygote during the early embryonic stages itself, it will get aborted. Okay, so yeah. A single protein that is part of a cascade of proteins involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female may transmit the disease to her sons. The possibility of a female becoming hemophilic is extremely rare because mother has to be a carrier and the father has to be hemophilic, and such embryos will not be viable in the later stage of life. The family pedigree of Queen Victoria shows a number of hemophilic descendants. She herself was apparently a carrier of the disease. That's why it's called the royal disease. That's about hemophilia. So you have two examples for X-linked recessive disorders, that is hemophilia and hemophilia and color blindness. Now we are going to learn about three more disorders, which are phenylketonuria, sickle cell anemia, and thalassemia. All of them are autosomal recessive disorders. You do not have any dominant autosomal or sex-linked dominant disorders in your syllabus. So phenylketonuria, I already told you about this as an example for pleiotropy. So this is also an autosomal recessive Mendelian disorder. This is an inborn error of metabolism. Inborn error means they're born with it, it's genetic. Okay. What happens here? In normal individuals, there is an enzyme called phenylalanine hydroxylase. Okay. This is produced by a gene. That gene is mutated in these individuals, so that enzyme is not getting produced. Normally, what is the function of that enzyme? This enzyme is involved in the conversion of phenylalanine, that is an amino acid. This phenylalanine gets converted to tyrosine because of the enzyme phenylalanine hydroxylase. Now that in these individuals where phenylalanine hydroxylase is not there, in the absence of PAH, this will get converted to phenyl pyruvic acid and some other derivative. Actually, tyrosine is very, very important. It produces many neurotransmitters in the brain. It is responsible for the production of pigment, melanin pigment in our skin. It acts like a precursor there. So tyrosine has a lot of important roles to play in our body. Now that phenylalanine hydroxylase is not present, phenylalanine metabolism doesn't happen properly. Tyrosine itself is not formed. As a result of that, skin pigment does not form. Such people will be very pale in complexion. Brain development does not happen because many of the important neurotransmitters will not get produced, and also because this phenyl pyruvic acid and its derivatives like other derivatives, they will start getting accumulated in the brain and cause mental retardation. Okay, so this gets accumulated and it's converted into phenyl pyruvic acid and other derivatives. Excuse me. Sorry. These get accumulated in the brain and that causes mental retardation. Since the kidneys do not reabsorb it very efficiently, a lot of phenylalanine and phenylpyruvic acid gets excreted through the urine as well. That's why it's called phenylketonuria. It's excreted in the urine as well. Yeah, that's about phenylketonuria. Now let's talk about a Mendelian disorder called sickle cell anemia. Sickle cell anemia is also an autosomal recessive disorder. So what happens here is, okay, so let's talk about sickle cell anemia. This is a condition where hemoglobin molecules are affected. In a hemoglobin molecule, you will have 4 polypeptides, 2 alpha and 2 beta. Within one hemoglobin molecule, you will have 2 alpha polypeptides and 2 beta. They will come together as and form that complex. Okay, hemoglobin pigment will form. In sickle cell anemia, the beta globin chains of hemoglobin are affected. This happens because of a point mutation. Any polypeptide is made up of amino acids linked to each other by peptide bonds, right? What happens in sickle cell anemia is the gene that is responsible for the formation of beta globin chain gets mutated. A substitution mutation happens, and in the beta globin chain specifically, at the 6th position, normally glutamic acid is supposed to be there. But because of the mutation that happened in DNA, at the 6th position, glutamic acid is replaced by valine. There's just one nucleotide base pair change. Normally, it is supposed to be GAG-CTC in the gene. Mutation happens, instead of AT, there is TA. Because of this one substitution mutation, this 6th amino acid gets changed. Okay, so instead of glutamic acid, you will have valine. What happens as a result of that, I'll tell you. Normally, inside the RBCs, hemoglobin is present in large quantities, millions of those molecules are present, and they're soluble in the cytoplasm. Okay, they dissolve in the cytoplasm. When people who have sickle cell anemia are exposed to low oxygen conditions, like they could be hiking on top of a mountain, as the partial pressure of oxygen and the air pressure overall reduces, there could be less oxygen available in their body in such cases, or when they're performing like strenuous exercises, they're not breathing normally, RBCs might not get sufficient oxygen. Whenever that low oxygen conditions happen, what happens is the glutamic acid present in the beta globin chain. Okay, so here you can see this one, this one, this one. Over there, the glutamic acid, it has a side chain that is kind of hydrophobic. It does not like water. Surrounding in the cytoplasm, there's water. So it does not want to come in contact with water. So what happens is one hemoglobin molecule and another hemoglobin molecule, whichever side has that sticky patch of valine, wherever, see, instead of glutamic acid, valine has been incorporated at the 6th position, right? So this valine has sticky end. It has a sticky patch which does not like water. It is hydrophobic. So the hydrophobic patch of one hemoglobin goes and attaches with the hydrophobic patch of another hemoglobin molecule that is defective, and it will start forming long polymers, rod-like rigid polymers. It will start forming inside the RBC. These rigid polymers are not soluble in cytoplasm. Hemoglobin, if they are like this, it is soluble. If they form these rigid structures, it is not possible, soluble. Okay, so they become insoluble. Now what happens is, normally RBCs have this biconcave shape. Because of the biconcave shape, they are able to squeeze in through capillaries and dissolve oxygen to all of our tissues. But because of this rigid shape, RBCs, rigid rods inside of hemoglobin polymers, RBCs lose their biconcave shape and become sickle shaped. They become very rigid like this. They become hard, rigid sort of pointed shapes. Now there's a problem with this shape. RBCs, when they're moving through these tiny capillaries, because they are biconcave, they can squeeze and move through well. If it becomes rigid and sickle shaped, they can cause clots within blood vessels and restrict blood flow, increase blood pressure, and do all of that, which can cause a lot of problems in our body. Okay, so that is sickle cell anemia. The mutant hemoglobin molecule undergoes polymerization, specifically under low oxygen tension, as a result of which the shape will change from biconcave to sickle shape. Okay. Now, what about the genetics and the genotypes of this? We'll understand that. So, there are two alleles. HbA is the normal allele. HbS is the allele for sickle cell anemia. This is the defective allele. Here there are three possible genotypes. These alleles or the gene responsible for beta globin chain is present on chromosome number 11. So this is an autosomal trait. It does not differ in males and females. It will affect the males and females similarly, unlike the ones that we've seen before, color blindness and hemophilia. So here, both the alleles can be normal. One allele normal, one allele defective. They will be normal, but they are carriers, or both defective, in which case they will suffer from sickle cell anemia. Now, this is true. These genotypes are applicable for both males as well as females. Okay, so if a person has to get sickle cell anemia, then both of the parents should at least be carriers, otherwise it's not possible. Okay, so here you have, it is controlled by a single pair of alleles, that is HbA and HbS. HbA, HbA normal, HbS, HbS disease phenotype, HbA, HbS appear unaffected, but they are carriers of the disease. They can pass it on to the next generation. That is sickle cell anemia. Next, let's talk about another blood disorder, which is thalassemia. Thalassemia also is associated with RBCs and hemoglobin, similar to sickle cell anemia, but there's a difference also. In sickle cell anemia, we saw that the globin molecules, beta globin molecules, were being synthesized normally, but the quality of the beta globin chain was not good. At the 6th position, ideally it should have had glutamic acid, but it has valine, which is causing some problems. But in thalassemia, that is not the case. Thalassemia is also an autosomal recessive trait. Okay, so here, alpha or beta, either of those chains can be affected. Here what happens is the gene responsible for the synthesis of alpha globin chain or beta globin chain, they are either mutated or deleted such that alpha globin and beta globin chains are not produced in sufficient quantities. If they are not produced in sufficient quantities, the hemoglobin will not be able to form a decent structure. The hemoglobin molecule structure will be defective. It will not be able to do its job, resulting in anemia. Individuals will suffer from anemia. The body tissues will not get sufficient oxygen. So that's the characteristic feature of thalassemia. Now, there are two different types of thalassemia, alpha thalassemia and beta thalassemia, depending upon whether the alpha globin chain synthesis is affected or beta globin chain synthesis is affected. The genes could either be mutated or deleted, resulting in that there is reduced synthesis of either beta or alpha globin chains. Let's talk about alpha thalassemia first. There are two genes responsible for the synthesis of alpha globin chains of hemoglobin. They are both located on chromosome number 16. The genes are called HbA1, HbA2. HB for hemoglobin, A for alpha. There are two of them, so HbA1, HbA2. So here, since we have two chromosomes, so you have four such alleles. Each gene will have two alleles because we are diploid. So you have two alleles for HbA1 gene and two alleles for HbA2 gene. Now, depending upon how many of these alleles are affected, the severity of alpha thalassemia will vary. If there's only one allele affected, of course they will suffer from thalassemia, a little bit of anemia, but it's not too severe, manageable. The more number of alleles affected, more severe will be the symptoms of the disease. Okay. Likewise, for beta thalassemia, the gene responsible for beta globin synthesis is located on chromosome number 11. I told you, right? Even here I mentioned that the gene is present on chromosome number 11. Here also it is beta globin chain that's affected. Here, if in beta thalassemia, it is the beta globin chain that is affected. So here, there's only one gene present on chromosome number 11. Since there's only one, we call it HBB gene. No 1 and 2 here. There's only one, HB for hemoglobin, B for beta. So, depending upon whether one is defective or both are defective, again the severity of beta thalassemia also will vary. Okay, again in alpha or beta thalassemia, unless both the parents are carriers, it's not possible for them to have children with thalassemia. Okay, so that's about thalassemia and sickle cell anemia. They're both similar, they're both different types of anemia. But sickle cell anemia is a quality problem. There's no problem in how much beta globin chain is being produced. It's produced in normal numbers, but the quality is not great. But in thalassemia, the quality is great. There's no problem with the quality, but the quantity, how much should get produced, that much number of beta or alpha globin chains does not get produced. So it is a quantitative problem. Okay, so that's about Mendelian disorders. Now we need to talk about chromosomal disorders. Chromosomal disorders are caused because of either less number of chromosomes than normal, or more number of chromosomes than normal, or an abnormal arrangement of chromosomes. Chromosomal disorders can form. Okay. So why does this happen? How is it that one individual can have 47 chromosomes instead of 46 or 45? That happens because during gamete formation, chromosomes would have not segregated properly. If they get segregated properly, we call that disjunction. Okay. But if there is any mistake during segregation such that one gamete ends up getting 22 chromosomes, the other gamete ends up getting 24 chromosomes, more or less than normal 23, then if that is successfully fertilizes the egg, or the egg itself is defective, whatever, if that is involved in successful fertilization, then the offspring that will form as a result will have chromosomal disorder. Okay, so this happens because there is a failure of segregation of chromatids during cell division, and that will result in either loss or gain of chromosomes. That is called aneuploidy. Ploidy means, number of sets of chromosomes. If that is in normal number of sets, like one complete set 23, two complete sets 46, and so on, then you call it polyploidy. Okay, then you call it euploidy. If there is one or two more or less than the normal set, then you call it aneuploidy. Okay, so euploidy is the multiples of normal haploid set. It may be monoploid or haploid, diploid, polyploid, like that. Aneuploidy means within the set there is more or less. Okay, so here in aneuploidy, you have two types, hypoploidy and hyperploidy. Hypoploidy means less chromosomes than normal. Hyperploidy means more than normal number of chromosomes. Hypoploidy, there can be two chromosomes less than normal or one chromosome less than normal. If there are two chromosomes less than normal, then it is called nullisomy. If it is one chromosome less than normal, it is called monosomy. If there are one chromosomes extra, then you call it trisomy. If there are two chromosomes extra, then you call it tetrasomy. So tetrasomy and nullisomy we don't see much in animals. Those embryos only will not survive. But we can see monosomy and trisomy. Nullisomy and tetrasomy, it is more common in plants than in animals. Okay. So we have to learn examples of trisomy as well as monosomy in this class. For trisomy, you have example of Down syndrome. So Down syndrome, you may have looked at individuals who have a facial appearance like this. Regardless of which part of the world a person is suffering from Down syndrome, all of them have similar facial features. They have a flat face with small eyes, and mouth is usually open, and the tongue is protruding, and they usually keep drooling. They are mentally retarded. So this can happen with both males and females because the autosome is affected here. So here, this is a case of autosomal trisomy. See, in our chromosomes, we have 23 pairs. The 23rd pair is the sex chromosome, XX or XY. That is not affected here. But the 21st chromosome, normally we have one pair, two chromosomes. Here they have an additional copy of the 21st chromosome. Because they have three 21st chromosomes, it will result in this disorder. Since the 21st chromosome, there's an additional copy, it is called trisomy 21 as well. What does Down have to do with this? It is not down up sort of a thing. Down is the name of the scientist who first described this disorder. His name is Langdon Down, and he described this in the year 1866. So what are some of the symptoms here? The individuals are generally short compared to the average population. They have a small round head like I showed you. They have a furrowed tongue. They have a partially open mouth like I said. The tongue is usually protruding and they have a crease in the palm that runs horizontally like this. And their development, physical development, mental development, psychomotor development is delayed. It is retarded. Okay, but they can live for a long time. They can live for as long as 60 to 70 years also. Uh, yeah, so that's about Down syndrome. Speaking of Klinefelter syndrome occurs only in males. Wait, before that, let me tell you about the karyotype of this. So, karyotype means what is their chromosomal complement. This can happen in males or in females. So, if it is happening in males, they will have 20. Wait, normally we have 44 autosomes. They will have 45 autosomes. Along with that, two sex chromosomes. That is the chromosomal complement. Klinefelter's happens only in males. Over here, this is a case of allosomal trisomy. There is an extra copy of the sex chromosome in males. Normal males will have one X and one Y, but these individuals will have two X and one Y, so there's an additional X chromosome. Since they have an additional X chromosome, they will show certain secondary sexual characters of females also. These individuals are sterile, meaning they cannot have children of their own, they cannot reproduce. I mentioned how they can sometimes show secondary sexual characters of females. One such character is gynecomastia, which means development of breasts in males. Overall, they will have masculine development. So they will have reproductive parts similar to males. They will have a pair of testes, but they are underdeveloped. They are not developed enough to produce sperms and reproduce, so they are sterile. Okay. Last one for the day, Turner syndrome. Turner syndrome occurs only in females. This is a case of allosomal monosomy. Monosomy means one chromosome less than normal. Here, normally females have two X chromosomes along with the autosomes, but these individuals have only one X chromosome, as you can see in the karyotype here. So that's why it is called XO syndrome also. So here, the karyotype will be 44 autosomes plus XO. Here, the karyotype would be 44 autosomes plus XXY. So, speaking of Turner's, people who have Turner syndromes, they're not too physically different from normal females. They might be slightly shorter. Some of them might show webbed neck appearance, but they appear mostly like normal individuals, not two distinct facial features or anything. The problem with them is that because they have only one X chromosome, their ovaries are not fully developed, they are rudimentary, they are not functional, so they do not produce eggs normally. Since they lack the ability to produce normal eggs, they will not be able to reproduce. They are also sterile. Yeah. And not just that, if there are not sufficient hormones and hormones produced for the production and release of egg, the hormones will not be sufficient to initiate any secondary sexual characters also. In females, you normally see secondary sexual characters where the voice becomes a little high pitched, there's development of breasts, widening of hips, and all of that happens. But in these individuals, you don't really see those changes. So that's about Turner syndrome. That's about this chapter. I have been yapping for the last I do not know how many hours. I don't know if you've understood all of this. If you've still watched it till this point, let me know in the chat. If you have any doubts, I will meet you again in the next class with yet another chapter. So until then, take care of yourselves, stay healthy, stay happy, keep smiling. Bye bye. Thank you.